So we have 8 minutes left of the tape.
This is one of these amphipods that you study went away. Yeah.
Looks like is this an acid floating there?
Mm-hmm.
Yeah. Or it might be something else
Do
caught
they
in there.
have really long antennae?
Not all of them. Some do.
Sit down. Oh, there.
Did you disappear the piece of paper? That was almost like in a ballot box. Poof. I voted for the ROV. What's that? What's that? It's just darker sediment, right? Okay.
Mm-hmm. How long was the transit? 799. I think it's 799. Yeah. Yeah.
Mm-mm. Shrimp.
Just the one I'm trying to do here.
Yeah.
You can try it, Jen. Just be careful when you come over this end here, look. Yeah, just be careful. Yeah.
Yeah.
Do that, Jen.
Bridge ROV.
RV bridge.
Can we increase to 0.4, please?
Increasing 0.4.
Thank you.
Hello. We collected a few amphipods from Galapagos.
Okay.
The species that Annalena described earlier. Yeah, isn't that cool?
Yeah.
Yeah.
So this is like a second report.
Second report, yeah.
Same area, was it more or less?
Oh, it
Bridge
was...
ROV, can we change the bearing to 315, please?
No, that was a little bit south of Iceland, I think. So, but along the same...
Ah.
Yeah.
Same kind. So it's blue transit.
Yeah.
Yes.
Just the very beginning.
Did you get lunch?
Yes.
That's good.
The soup is very good.
Mm.
The fish is okay. And there is chicken.
I think the fish is going to be lovely. I miss eating more fish. So that might be good.
So we didn't say that we were transiting the...
I am putting in the transit and I have put in the... yes, we don't have the start here
Yeah.
and we don't have location for that one.
But we have these.
Yes, and then you have to remember to put in... You have to remember to put in the start of the transect here with
Okay.
data.
Yeah. Genau.
5 minutes to tape change.
Okay.
That transit will be interesting because we are going really up.
Yeah.
Mm-hmm.
About 500 meters, something like that.
And that will mainly be on the next shift.
Yes.
But that's okay. We can sit in the dry lab and watch.
Yeah.
People who are watching us live can just continue to be in the same place because very soon we will be on the next transect that has been planned by Julio, who is here to take over from us really soon now. And he has planned a really exciting third transect for the day. What is this? Can you zoom in on that? Are they Seriantids?
Blue transit. Forever.
Yeah.
I think they're Seriantida. So these are anemone that like to. They have a tube under.
Yeah, that's first time that we see them.
Yeah,
Today, right?
cool. They are large. Look at that.
Yeah, indeed.
Yeah. Seriantida, and I think now we are getting those little sponges. So this is a different
Yeah.
soft bottom community than we have seen until today on this dive.
Yeah, indeed.
Because all of a sudden we have the anemone, the tube anemone, the Seriantida. No, it's okay. And we are seeing maybe also some of those. Maybe I'm just dreaming, but yeah, look at those. We have some white dots there.
So,
No, they are
not those.
no, but we are seeing Seriantida.
I'm gonna leave the notification like this.
Yeah, that's cool.
We're going for.
Francie will be back.
Yeah.
She's just asking if we should keep streaming the transit.
Yeah. That's fine.
That was nice to see. That was really cool. A little
Yeah.
bit of new community there.
Ending with new stuff.
Yay, that's always good. We're ending on a high.
Do you wanna take over?
Yeah, go because otherwise you won't find anything.
You come with me?
I will stay until Francie comes, that's okay. So Julio is taking over for Sergi.
Oh, it was great.
So this is Francis.
This is me.
Thank you so much.
This is nothing,
Bye.
right?
Welcome.
Yeah, this is transit to the next transect. But we saw the cool Seriantida, which was good.
Do you want me to
So
focus?
let me just move this here so you have enough space.
Bye.
Enjoy lunch.
There was a Seriante?
Yeah, several. There was a field of Seriantida.
Are those shining bioluminescent?
I am not sure. We do know that other anemone sometimes are bioluminescent. Actually, in the deep sea, quite a lot of the animals would be bioluminescent, but many of them are mainly visible bioluminescent when they get a little bit disturbed, or if they are, like hitting a screen or something like that. So there are several different reasons for being bioluminescent, either to attract food or to scare away enemies or to inform enemies that you don't need to eat me, or maybe just to hide. There's this really cool thing that some animals are doing to become almost invisible, so they are using counter shading, so a little bit higher up in the water column, around 1,000 meters, your stomach can maybe have a little bit of bioluminescence, because if your predator is sitting under you and wanted to eat you, you are looking just as light as the light coming down from over you, and then you become invisible in a way.
Everything is invented.
It is so cool. I always wanted to have Harry Potter's cloak of invisibility, and that's what they do have.
Really, a little bit shiny on your bottom.
I would like to be shiny in my belly. Shiny belly would be nice,
Shiny
huh?
belly.
Okay, 1 minute until tape change.
What is this?
If you're going into the blue transit,
Yeah.
if you're going into the blue transit, we will just join the video, and the video will stay on, but we can stop the recording.
So,
Yes.
so we will have a shift change in tape change, and then a shift change in about 20 seconds. and then Franzi will take over for me.
Actually, we would probably go in the blue transit now.
And then we might go into blue transit to the next transect, because Julio has decided on a really cool next transect. But now we should change the tape, please.
Yeah.
Yeah.
1, 2, 3.
Stop.
A bit too far away, maybe.
Yeah. How long do you think the blue transit will be? So it's like 800
Meters.
Yeah. Unless we found something interesting in the way.
Probably
It's like this.
45 minutes to an hour,
Yeah.
Yeah, okay.
roughly.
Yeah.
Roughly.
But we will
That's the
continue, yeah, we will continue recording, right? We are continuing recording, right?
Yes.
What about the stream?
The screen will continue, but they will have new places. So we will treat
Sorry?
Reach ROV.
Right, RV.
Can we change the bearing to 325, please?
Changing to 325.
Okay. 1, 2, 3.
So we started the new tape, and I think what was happening is we are going into a blue transit, so we will lift the ROV a little bit off the seafloor to be able to move slightly faster toward the next transect that Julio has been looking up for us, designing,
Yeah.
picked out from all of the wonderful bathymetric maps that are already there. And we will keep the live stream going, but if you need to go and get a cup of coffee, this might be the time to do it. Because we will, of course, you will see the video all the time, but we will turn off the sound now for a little while, so you don't have to listen to us chattering.
Yeah.
And it will probably take 45 minutes, maybe a bit more.
Yeah, so check in if you're super excited. Maybe check in 40 minutes. Start tuning in and have us in the background, and you will hear when we get to the next new place, right? Yeah.
See you soon.
See you soon. Let's do it.
Okay.
Thank you.
Normally the dry lab, you should be able to hear us now.
So we're pretty much. That's the ship. Yeah. Right. Let's keep going on the whole that line.
Dry lab, can
That color. Right.
the two red dots on the microphone, can you hear us?
Shift cam.
Hear you all loud and good, thank you.
Shift cam is on one. Right.
Thank you.
So I can take care of this. You take care of the annotations.
Will do. Tell me when you're ready.
Yeah,
Okay.
and zoom and stuff. We're ready?
We're ready.
We're ready.
Okay, let's do this.
Let's lift off.
We're now on our third transect, 2,548 meters.
And we are a crustacean expert. Can I call you a crustacean expert, or is that more specific than that?
You may. May I say that's a sponge expert, Paco?
Absolutely. And we have Andrea as our pilot,
Hello.
and Patty,
Co-pilot.
co-pilot.
And we have white sponges, one rocks.
Yes. So we are still climbing on the little hills around Moon's treasure, uphill.
What are these sponges?
Amphidiscelamonae. Where is the book? The tulip one. That's on the left side.
So a
Or
tulip.
you can write tulip glass sponge. Yeah, yeah.
Tulip. I may zoom in a little bit.
Yeah. Yes. Again, the tulip sponge here, and Asconema mega atrialia, those vase-shaped brown glass sponges that we see here and there. And for those who have seen our previous dives, we see a little gray sponge that's covering a lot of the rocks, and we're trying to put a name on it. So
There's a fish.
that's a fish.
Recorded. Good
Yeah.
idea. Hello, Grant. Visiting us.
Would you like
Which
to
is
see
nice.
this?
I would like the file from the transect start stop file.
This one.
Thank you.
Mm-hmm.
Do we have an extra sheet for? No, this one. We should. We have.
That's all, yeah.
Yeah, before onto here, please. So this is our start.
This is T3,
This is T3 with
same dive,
2,540 meters now.
21st. So we started at
Lots of
220.
sponges.
I do.
Yeah. This is sponge country again.
Yes. Rolf, can we slow down a little bit, please? Then the viewers don't get dizzy when we zoom in.
Crinoid on
What's on it?
and something.
Crinoid and a decapod.
Yeah.
First I thought it's a sea spider, but it's another crinoid back there, also on the sponge. So they're trying to get higher in the water column. So they climb up on the sponges or on any hard substrate they can find. Because the current is slightly different from directly on the seafloor. And they grab the little particles out of the water. Oh.
Do you need more time, Anna?
No.
No?
We may continue, please.
Okay.
Okay. That's filled.
Can I grab
That's
that
an
one
amp-
that you just completed, that piece of paper? And I'll bring it back.
Sure. So a lot of the what I'm calling now the pom-pom polar sponge is those white bushes. There's a lot of them here.
Hi guys. Hello.
Hello, Jenny.
Hello.
And this is typically a volcanic rock field. And apparently sponges love it because there's so much hard substrate to overgrow. So if you were looking for your animals, where would you look for in this type of habitat?
Well, some of them sit on sponges.
Mm-hmm.
And, well, I'm working with amphipods. Some of them sit inside sponges, so if you take them apart. Lots of them on soft sediment. But they have all kind of life forms. So if you have like the scavenger guild, you'll find them when there's something like a dead fish or a whale falling down.
Mm-hmm.
So they're attracted by the odor, the
Mhm.
smell. And
Do
then
you
you'll
know-
find lots at once.
Do you know if they eat dead sponges?
I am not sure, but some definitely eat sponges. I'm not sure because they have the spicules of
Mm.
the sponge in the stomach.
Mm-hmm. Can they live in the crevices between the rocks as well?
For sure.
Yeah?
I think a lot lives between those rocks and
Mm.
crevices. It's most of this life here is so small that we can't see it immediately.
Mm.
And all these wonders occur when you look through the microscope in more detail. Are these crinoids again? Coming up on the left.
Yeah, with some shrimps on the left as well.
The red things floating around are shrimp. And the brown parts
And
are
in the
aqua.
middle, this piece, what looks like a piece of wood, is a stem from a glass sponge that probably died, but the stem stayed, and it will disintegrate, and the spicules will probably fill the crevices here.
Is that Aspergillus? That one?
That is, yeah, it's probably fallen, yeah. Yeah. I mean, it's probable that sometimes you have landslides and maybe they take up and make the sponges fall. I don't really know.
Well, Paco likes to point out how old sponges are. They are the oldest, but I have to say that crinoids are also super old. They're also from the time of the dinosaurs.
Yes, that is true.
I know.
But sponges are one of the oldest because there's still a huge debate in the scientific literature between is it ctenophores or is it sponges that are the first animals to emerge on our planet? And it's been a debate that's been going on for more than 20 years, and we still don't have a clear answer. So every year there's 1 or 2 papers that say yes, another paper says no. So it's still very unclear, which is really interesting, I think, because if sponges appeared after, that means they probably have simplified their bodies secondarily in evolution, and that is very interesting.
It is. There is a debate of the origin of life, and it started in the ocean for sure.
Mm.
And most likely, or very possibly, at some height of thermal vent sites. And here we are only 30 kilometers away from the active vent sites that we'll visit next days. This is an inactive site, which has been discovered, I think 2002.
Moon's treasure, you mean?
Yes.
I don't know, actually. Do you know, Jenny, when Moon's treasure was discovered?
No,
I
I'm just thinking.
I think it was 2002.
I talked to Anna Helene about it yesterday, and she said that she remembers very well the discussion about how to name this site when they first discovered it. But I forgot.
Look at this little amphipod.
Oh,
Is it an amphipod?
yes, it is.
Wow, a big one.
It had such a red stripe in the middle.
No, I didn't ask her myself
Okay.
when it happened, but oh, there's another one.
Yeah. So those little red.
Oh,
There's a shrimp or a mysidae. I don't know how to recognize them.
A decapod.
Or decapod. Yeah.
Photopod.
So right now, with my microscope, I'm trying to identify these gray patches of sponges that you find on all rocks. I've been drawing the spicules and looking through the literature. Can't find it now, which is getting exciting because it's so common, but maybe it doesn't have a name yet.
oh yeah. I'll just zoom in a little bit. Maybe.
Wow.
Have you got a name for the fan-shaped sponges?
No, but we collected it today, so next time, until next time, we might have a genus name, and then we'll take it from there. But at least I really want to know from which group of sponge they belong to. They don't have a name yet.
It's a stalked crinite.
Oh yeah.
On the right side.
Yeah, yeah.
Speaking about crinites, they come in. There are two main shapes. The one with the stalked crinites are also called sea lilies, and the other ones are the feather stars. The latter can move around. They can swim. Oh, what's that? Sorry, too fast.
This
Look,
looks very
This
steep.
goes up. What a pretty amphipod.
Oh wow.
Come on, go right for the lens. That's a nice one. Come on, sit. Can we stop, please? I would love to. Can you stop,
Yeah.
please?
Do you want me to stop?
Well, I would love to have a picture of the amphipods. If it sits down right in front of us, I would be.
Yeah.
Oh, what are you
No,
doing?
but because it's zoomed, if you
Sorry.
wanted to move less, yeah.
It's too fast for us.
Yeah.
Wow, this is beautiful.
Now
Mm.
we'll have a little plume, which will settle quickly.
Wow.
You're sitting a little bit
right?
So
We are stopping
Yeah,
a little bit.
okay.
We'll stop a little bit and have a play what we can discover by
And-
zooming up close.
yeah, we can enjoy the view as soon as it's cleared out. So this dense accumulation of sponges can be named depending on the community we find in them. Usually when there's 2-3 key species of sponges, we name it a sponge ground. If it's more diverse, like it's the case in Antarctica, for instance, where there's sponges, very dense accumulations of sponges, but a lot of different species, then we talk of sponge garden. And here, since we don't really know all the species, that's something we will have to investigate. Is it a sponge ground? Is it a sponge garden? So far, it seems to me that there's not a huge diversity of sponges, so it might be a sponge ground. So here we have a close-up on the polar pom-pom sponge. Ooh, should we zoom there?
Yeah, just a second. I wanted to get the overview first,
Okay.
so what other animals we can see. Yeah, no, okay, move there, please. So, oh, good.
Yeah, so the brown sponge, I think, is Hemigelius arcofer. It's a species we've identified here on the boat. It's been described a long time ago, but we had never seen it alive. So actually, for our viewers here, you're also probably one of the first humans to see that species alive, pumping, filtering the water. And that's pretty exciting.
I'm not quite sure. It's hard to see. Is that an anemone?
That's an anemone, I think,
It's-
yeah, on top of
well,
it. Yeah.
no.
There, yeah,
There.
yeah.
I'm actually surprised.
Can you
that-
zoom in a little bit more?
Nope.
No, you're maximum. Okay.
Next. I'm surprised that there are so few bottle stars and sea cucumbers, because quite often in this kind of terrain you'd see them when zooming
Mhm.
in.
So here again we have
So scroll to the
Hemigelius arcofer, but this time a much more developed specimen.
Would you mind? This is
Yeah, it has produces these fibers that come out of it, giving it the impression that it's very hairy, like the woolly mammoth. we could call it the woolly sponge.
wow.
It does look lovely, and
I
you
really
see
want to touch it.
you'll have some spicules.
So again, it's the
But
first
look at
time.
this. So I'm trying to zoom in on the soft coral actually.
Oh.
So this is not a sponge, the left parts.
Yeah, we have colonial Ascidians, soft coral, and the woolly sponge.
How's it called again?
Hemigelius arcofer.
Okay.
And I didn't know that last week. So it's because we collected it, and with Julio we've been trying to find a name for it.
Yeah.
And we finally found which species it was the last few days. And now we see it everywhere because it's one of the really common ones. So that's one advantage of exploring the deep sea with a robot, is that we can start to put names on things we see down there.
Yes. We may continue.
Okay.
Yeah.
Thank you.
Sure.
I better load my soft coral. And Ascidian.
And woolly sponge.
I think that's one of my favorites so far.
Mm-hmm.
It's really cool.
Yeah, it is.
The name is not though. I have like I need to write it down. I
Yeah.
have,
Yeah, I can write it
Yeah.
down and show you later. Yeah.
Wow.
There's the tulip sponge again at the bottom of the screen. It's a glass sponge with a stem. Very beautiful and delicate sponge.
So most of the life you can see here is filter feeding or suspension feeding.
Yeah, absolutely.
And all this nutrition comes from the top, falling down. Oh, let's zoom in. So there's an anemone.
I think. Like
Another
this one? Or was it smaller?
No, it's all good.
Okay.
Another one. Some more crinoids.
So many.
Yeah.
Mm.
Wow.
Crinoids have this many arms, and like most of they're related to like sea stars or sea urchins. And they can regrow their arms, so when one breaks off, it regrows. It's a nice ability.
Do crinoids have predators? Do you know
If they have
Well, I
predators.
guess sea stars, because in the previous
Oh, yeah.
dive they saw sea stars. But the crinoids that are not stocked, without a stem, are they also eaten by sea stars? Do we know?
I'm trying to think if I've seen an image. I haven't.
Because that's a
Eaten by
question we
fish.
often get: is who eats who in those environments?
Yes.
Who's the apex predator? Because there's no tyrant, there's no dinosaur, there's no lion. So who is at the top of the chain? What do you think?
Well, if you go to the very deep, it's amphipods. I have to say.
Amphipods.
Actually, don't you laugh, because if you go down the deepest trenches, you know,
Okay.
below 6,000 meters, if you go down to the Hadal,
Mm-hmm.
the fish can't cope anymore. Or very occasionally they do. And then you get the large amphipods. So I often show them as
Okay.
top predators.
I
So
have
like,
a di-
it's like a lion in the savannah. It's
I have
in the middle.
another possibility for you. I would say sea stars.
Nope, they can't cope so deep.
If an amphipod battles with a sea star, who wins?
The amphipod, of course, the seastars can't even cope when there's no salt. You know, they give up in the Baltic. So they need a very salty environment. And amphipods, on the other hand, occur also in freshwater and in caves.
Okay, so they're more adaptable. You're
So,
saying.
2 points, crustacean. Yeah? Okay. They're more adaptable, and they have adapted everywhere. But this is sponge heaven.
It is. It is, and again, I'm saying that this type of habitat I have never seen before. We think it might be a new type of habitat that should be described, but for this we need to know which species are present. We need to understand the community that we're seeing. So this morning, I know that my colleagues have already collected a handful of these sponges, and we will continue to do so.
Do you think these thin fan-shaped sponges are a different species than the thick fan-shaped sponges?
Yes.
Okay.
Yes, and I know they've been collected this morning, so I hope I will have a name or a genus name for you tomorrow. But right now they've been people have been taking pictures, previous expeditions, Norwegian expeditions, so they've seen them, but they didn't collect, so we don't know what they are.
Okay.
So I'm very excited when the robot will come with those samples.
Okay.
Tonight, probably. I'm very excited to look at these.
Many of those sponges will reproduce by larvae. So
There's all types of reproduction. So some will have larvae that will swim away and settle somewhere else. Some will release little babies, already formed little babies. And many of them will just send their sperm and oocytes in the water. So fecundation will take place in the water column. And then it will produce a larvae again that will be taken up by the currents, water currents.
the same is true for the crinoids, or for many specimens that we see here. And often these larvae need a hard substrate to settle. So when they're looking for a landing place, it can be a rock. That's also why this habitat is so super suitable for them, because you have all this hard rock scattered amongst the soft sediment.
Absolutely. But I don't know if you've noticed, a lot of sponges are very clean. There's nothing growing on them. And on the other hand, you have some sponges that are very social in a way that they attract a lot of other organisms, and they build these whole little communities. And the difference between these two types of sponges is still poorly understood. We think that some sponges are happy to have other animals growing on them for protection against predators, maybe seastars, maybe amphipods.
There's a woolly one again. There was a woolly one again.
Right there at the bottom?
Yes.
Yeah. Yes.
There it
Yeah.
is.
And the anemone on the left.
And the sponges that are clean probably produce a chemical to deter animals from sitting on them. And these chemicals are very interesting to us because we could actually reuse them, maybe to make products that will deter also organisms from sitting on the surface. And I'm thinking, of course, of the paint that you use for boats, for boat hulls, because currently we have a lot of paintings that are very toxic to the environment. But if we could find a more natural compound, especially coming from the sea, that would be very interesting. So there's a lot of research groups in the world trying to extract, we call them anti-fouling compounds, for applications, for instance, for the ship industry. And many of these anti-fouling compounds are coming from sponges, because you can see how clean they are. They clearly have found a chemical to
Can we stop, please?
clean themselves.
Please. Can we stop, please? Thank you.
I can already hear you getting very excited.
I am excited.
And
I love
see
seeing
why.
this live.
I see why.
So please enlighten me. I don't see why. I see the sponges.
It's a sponge.
Oh no. This one? This little guy?
No, to the top right. Sitting. Sorry. I'll show you. It's right in the middle. Now turning at 6 o'clock. It's a decapod. Turning up.
Yeah.
It's upside down.
Yeah, I see it now. Yeah.
It's quite cool.
I clearly don't have an eye for these small crustaceans. Not yet.
You can see the eyes glowing and
Oh,
the long
there's
antenna.
an outfit joining the party. To the
Yes, but
Read your reveal, stop please.
But it's swimming along. It's very hard to see when
it
they
might
don't sit
settle.
down.
Come on, buddy.
now that we talked about larvae.
It's actually looking at us. You see? It's towards us. It's looking at the camera.
Yeah, I'm focused
For
on the
sure.
alphabet.
Yeah. It's the alphabet I'm talking about.
It's
Yes.
set down.
Oh, now you finally sit down.
Yes.
Try to do my steering a bit to the right.
Yeah.
It's actually a good question why they have eyes, because we're now in 2,500 meter depth. We don't know. Some of them. Oops, stop! I'll try and zoom. I'm zoomed out. Some of them stay down in the dark all the time. One assumption is that there's a lot of bioluminescence going on and illuminates parts so they can attract partners or irritate predators or attract prey themselves. There are many theories.
Would you like to try to collect that amphipod?
Nope.
No?
We're too far.
Or too far?
We're too
Oh yeah,
far.
now I see. Yes.
We're too far, and the arm of the robot will not go that far. Maybe continue. Okay, thank you.
This way. Ship on first. Great, Jarvi. Can we resume the move, please?
Resuming move.
Well, we talked about larvae before, but the critters that we just saw in red, the shrimp and the amphipod. Well, it's actually only true for the amphipod. They don't have larvae. They have a brood pouch, and they take care of their young.
So it's the
So
mother
the-
that takes care of the young?
Yes. Yes. This question might sound a bit bizarre because often in the animal kingdom it's the mother, except for the sea spiders that we encountered yesterday or the day before. And it's a dad. We're now waiting for the ship to catch up with the RRB. We might as well zoom then. Yeah.
So on the image you can see two little red dots. These are the lasers coming from the robot. And they are 20 centimeters apart?
Yeah, 20.
20 centimeters apart, so that gives you, in the audience, a feeling for the size of things.
Like this critter.
Which one did you see?
Or just out. I'm really surprised that we don't see any turtle stars anymore. None.
Look, sorry, I just wanted to look there.
Okay.
Ready? Off you go.
Yeah?
Yes, we're ready.
Yeah. So, bridge RV.
Raised.
Yeah, we get a bearing changed to 310.
310.
Thank you.
So it's very tricky because we are connected to the boat with a cable, and so the boat is moving with us, following us, and we constantly, I mean the drivers here have to constantly tell the boat to either stop or move faster or move slower depending on what we're doing with the robot. So it's
I forgot the English word. What's it called? An
umbilical cord.
umbilical cord.
Yeah, kind of.
But it is.
Like the mother to a child.
It is. It's
And here in the ROV, in the robot shack, where we control the robot, we are actually sitting on the boat. We are in the container that's on the boat. So, oh, jellyfish.
Yeah.
Raised. Paddy, just to confirm, you want speed 0.1.
Yeah, thank you.
There's another tulip sponge on the top right. This globular glass sponge sitting on the stalk.
That's the big round, the donut-shaped thing. The donut-shaped sponge. No. That one? Is that the
That's what
tulip?
I've been. Yeah, the tulip. Yeah.
That's big.
Yeah. Yeah.
You have very strange tulips in your country.
Oh, now I see it. Sometimes the perspective is funny.
Mm-hmm.
It looked a bit like a donut for like a couple of seconds, and then it changed to
Are you hungry, Jenny?
no. But actually, Francie and me were talking about donuts at breakfast, so maybe I'm a bit
Influenced. Yeah.
influenced there. Yeah.
So here the polar pompon is everywhere. And yesterday we got a couple of specimens from that interesting species, and we found that it was reproducing. So our researcher Sergi was looking under the stereo microscope, and he started to see things inside the sponge, which could be eggs or larvae in development. So that's very interesting, because sponges, like many other animals, have a reproductive stage. They don't reproduce all year long, because reproduction requires a lot of energy and food. So,
But we don't yet know if this is coupled to a time of the year, or if this
absolutely.
is year-round, because there's so few samples that it's always just a glimpse
Yeah.
of
And
chance discovery.
We should collect all year long to be sure to actually pinpoint to which season they reproduce.
That is true for all organisms that we see down here.
Ask the name.
This one?
No, to the that is now moving left in the.
Ah, there.
Yeah.
Yeah.
Megatriana.
Yeah.
Thank you. 2 weeks into the cruise and I got it.
The name comes from atrium,
Okay,
but huge atrium. Atrium is like a vase shape.
right.
So mega atrialia,
Ah,
which comes from atrium.
that's why the double A. Megatrialia.
Yes, exactly.
Ah.
Yeah, I should have explained that to you when I gave you the name. But it's a good way to remember it.
Now it's.
It is a
It
good.
will stick with me forever. Megatrialia. And now it. Paco, this was the
Well-
key to.
We're learning lots of new names
Yeah.
from each other. Most of the deep sea critters don't have English names because they're so uncommon. And our job as taxonomists is naming new species. Oh, there's a jelly.
Jelly.
Oh, our tape has to be changed here in 10 minutes.
I really like polar pompon though. That's a funny one.
We've been discussing this with my sponge colleagues.
We'll just remember, we'll keep it in UTC yet.
Okay.
Go, do you want to stay? And because the tape has to be changed. Okay.
I can do some tape.
What do you need?
No worries. I can bring it up.
Oh,
I can do it. It's 10 minutes, man.
Yeah.
You're alright.
Well, the screen where I'm entering just went black. So it's like no. It's come back again.
Sorry?
That's the blender. Comes down.
There's the woolly-
It's woolly sponge.
This is the back.
Ah, nice ones, eh?
Vaticalis.
Those.
Deep sea shrimps.
Yes.
So as you can see, the polar pompon, they are easy to grab and collect, but there's so many other little sponges sitting on those rocks that are really
What's that? Is that
Tiny,
a sponge? Can
tiny.
you spell to the left? Left. Thank you. That's an anemone. Upside
there's
down.
the woolly sponge next to it.
Yes.
Yeah, so a lot of these little crusts on the rocks are sponges as well, and they are complicated to collect. So usually we take a small stone to be able to collect it, because otherwise it's impossible to scrape those stones. Here's another woolly sponge.
Yes, definitely.
Very nice.
There was something going on.
Quite
I think
steep.
that one had a parasite.
Yeah.
It really looked like it.
Mhm.
Against the current and like keep it moving. As you can do, so fine with the current line.
How easy is it to stop to collect on such a steep? It's possible.
What do you want to get?
I'm just thinking ahead to know if I tell you to stop, it's possible or not.
I'm just asking because if it's too close, I don't know if the slurp would be too close, but
Mhm.
we can
Because of
try
that-
or
because of that catch?
It's okay. Okay, it's not too bad.
Okay. Thank you. Wow, it's so beautiful.
Yeah, it's incredible.
And I mean, you would not think of a better polar scene as these white sponges everywhere, like snow on rocks.
Look at the tulip to the left.
To the yeah, absolutely.
Really cool. Wow.
Wow.
We really have amazing pilots here too, huh?
Mhm.
Flying this slope, just so slowly
It
and
is steady.
steady.
So smooth.
Amazing. Thank you.
Climbing up the hill.
Yeah.
Running up that hill.
Running up the hill, exactly.
It's just amazing.
It's hard to see on a camera in that angle, but this is a very
I know,
steep
because
slope.
I've noticed.
Yeah. Keep doing your bit, man.
I started with 204. So
Almost everything that we can see here is a sponge.
Yep. But the big difference with the top of the seamounts that we've seen previously is that there is no spicule mat made of the dead sponges. Here it doesn't seem to take place. And that's interesting as well. Here it's just rocks and the sponges sitting on them. While at the top of the mountains it's sponges sitting on dead sponges, on the remains of dead sponges. They've created their own hard bottom.
These were crinoids again.
small underwater cave.
How many fan?
Mm. So we are at 2,481 meters. Wow, look at the amount of small sponges and feather stars
Excuse me.
and shrimps. Oh, this is very rich.
Turn off the lasers just for a short moment.
Okay.
Okay.
Should I zoom out a little bit or?
Okay. Well,
Yeah, like this,
zoom
yeah.
out,
Oh.
and then we'll stop.
Stop.
Yeah, yeah.
Yes,
If you
please.
can go down a little bit to see the.
Okay. There's some plume
Ah.
chasing this bit.
Okay.
We can wait it a little bit.
It's quite interesting how they go on the edge of
Yeah.
the rock.
Yeah.
Obviously, the best spot
Yeah.
for the.
Radio,
Okay.
all stop, please.
It's a good moment to change the tape.
Yeah. So we do that.
Yep.
Okie dokie. So we're going to stop in 3, 2, 1. Stop.
Tell the dry dive guys.
Yeah, we're changing the tape.
Shall we tell Jason? Mike?
I think we are hearing
They can hear us well.
Ah, beautiful. Why are you laughing? It's
lots
a-
of colors.
Yeah, yeah.
It's very nice.
It's a good thing the crustaceans are there. They bring much more colors than the sponges in this area.
I think
Okay, recording on mic on in 3, 2, 1.
it's true for many situations in life. Crustaceans bring more colors. Oops. As we said, these are filter feeding, and they have some mucus on their arms, on the feather stars, and little small cetae, and they're catching anything they can. So, say plankton, but also eggs, or even tiny fish.
Mm-hmm. Can I zoom on the shrimps?
You alright, John Danny?
Yeah.
So what do they eat?
If
The
you
shrimps?
look at these feather stars, how they're stuffing the. They've got this one opening in the middle, and when they caught something, they stuff it in, so you can see the arms going into their mouth,
Mm-hmm.
not like sucking the units off,
Yeah.
and then releasing them again to catch more. You see the left one there? It has nearly all arms in. So
Yeah.
she
Absolutely.
just had a feast.
Mm-hmm.
What is this wormy thing here? There's a white thing under the
Yeah. I can.
crinoids, which we can't see properly. Oops. And you also see. You see all this orange little units on the rock.
Worms.
I assume they're
Yeah.
tubes of worms.
Yeah, tubes of worms. Oh.
Yes, right?
Look at me.
Yeah, he
That's
was
right.
jealous.
Like a dog paddle swim.
So we, in the previous dive, we have collected those hanging round-shaped sponges, and very interesting. They produce a lot of mucus, a lot of gooey texture, kind of viscous texture, and we don't really know why. But they produce a lot when we brought them on board.
And that's a real nuisance if you have other samples in the same pot, pottle.
Yeah.
Because we are also doing DNA extractions, and what you don't want your clean, lovely little animals to have mucus all around them. Because when you then do the sequencing, you sequence sponge mucus. That's what we're trying to avoid. So that's why we're also sampling in different units. Can we zoom out again? A little bit to the left, please. This one. What is this?
The stick, you mean?
Yes.
I'm not sure.
I don't know either.
Maybe remains of a stock, actually of a sponge stock, probably.
That's what a sponge. That's what I think as well.
Yeah. So again, these gray patches that you see here are sponges as well. We call them encrusting sponges. They are little crusts, so they don't grow much more than that. They can expand on the surface. And this particular one we have collected, and we're currently trying to put a name on it. And we've failed so far, which is kind of exciting because maybe it's not been described yet. But we'll see. We will continue to. We will update you on what this species is.
Down here, it's mainly sponges encrusting. In the more shallow regions, you also have the encrusting corals. And then in the photic zone, so when there's still light, you have the encrusting algae. Often all competing for space. But down here, it's sponges only. Zooming out.
So what do you think the shrimps are doing? Are they just picking up stuff on top of the sponges, or they're just sorting and eating really tiny organisms? Or
I have to disappoint you. I don't think they really care about the sponge.
Okay.
I think they just use it as, well, wherever they can sit.
aha, so they're actually eating in the water column?
Yes.
Aha, okay.
Filtering. So
Okay.
shall we fly on?
Yeah, we can fly off. Oh, look at these fantastic volcanic rocks with clean cuts. This is basalt for most of them, and you find basalt on the upper part of the oceanic crust. It's the magma that solidified really rapidly in contact with the seawater.
Photograph from the back.
Oh, beautiful crest on the rock.
Where? Oh. Mm. Yeah. Right. The sponges are smaller here.
They are.
Mm.
But there's still a lot of encrusting ones. We're now at 2,480 meters.
Bless you.
Thank you.
You see a little red jellyfish
Yeah, little red jellyfish
swimming in.
on your right. Oh, and the second one.
yeah.
Or yeah, that was different.
It was definitely something different. I'm not sure if it was a jellyfish.
Oh, but look at that one.
That one.
Beautiful. Ooh. Oop. It was a shy one. It was very shy. It seems to me that the crinoids are also very social because, I mean, several could live on another neighboring rock, but they like to be close to each other.
Well, the crinoids are the feather stars.
Yeah. I think that's also a glass sponge stock, maybe from Colophacus.
With the feather star just crawling up.
Yeah. It's taking advantage of the remaining stock.
Well, feather stars occur in all oceans,
Mm-hmm.
especially actually in the tropics. And they're around 600 species. How many sponge species are?
10,000?
Yeah. The same as amphipods, by the way. I'm not making that. It's also around 10,000. So on our internal battle here.
Two.
Well, I think Paco wins here in this territory.
No, but I mean the number of mollusk and crustacean cannot. I mean we cannot match that. They're super hyper diverse compared to sponges.
Yes. But you've got a phylum.
Yeah,
And I have an
we
order,
do.
so it's
But I have to say this
This-
as well, is that 10,000 sponge species described, yes, but probably that same number of undescribed still to be discovered and described and named.
that is not only true for sponges, of course. This is true for all these groups. That the number of undescribed at least matches the undescribed.
And that's why we're still finding new species in those habitats, especially here, which is pretty much unexplored. Here we have a beautiful round rock. This is a typical pilolava. So you will find pilolavas on top of the oceanic crust, and they're typical of magma coming up and solidifying super rapidly when they meet the water. And that's how you identify old oceanic crust. So if you wander off in the Alps, you will find pilolavas, because before there was an ocean between Eastern and Western Europe, and this ocean closed up, and the oceanic crust that was in the middle was mixed with everything else, and now you find it on top of the Alps. You will find pilolavas, indicating that there was a sea here before. So how long have you been working with amphipods?
Oh, ages.
Was this but from the start?
Can I say last century?
Yeah.
Sounds terrible. more than 20 years.
Did you do your PhD on amphipods?
On groups of crustaceans, actually in Greenland.
So
So
not-
masters, and then my PhD on Antarctic
Uh-huh.
stuff. Mainly amphipods, true.
How many species have you described since?
I have to admit I have not counted, but around 50, I guess.
Around 50. Okay, you have to check.
50.
You have to check your homepage on Wikispecies. You should have your author name there, and maybe someone did that count for you, because they're really good at that.
It's my lack of vanity that I don't know the species. I do know how I would potentially check that.
But yeah,
you can
I'm
also-
telling you, probably you
Yes,
have a web
no,
page
page.
on Wikispecies,
I have
and,
not checked.
and people write
Wait.
the new species you've described, and then you can
Okay,
easily count them.
When
Bridge, are we?
we look into worms,
Yeah, can we continue the
The
move,
worms.
please? Point 1.
and
3101051,
Thank you.
it's a fantastic webpage, the World Register of Marine Species. And if you have doubts of name spelling, or when things were described, or who described it, and what publication, it is great to look.
Mm.
Also getting more features and where things occur.
Yeah. So it's called WORMS, the acronym of World Register of Marine Species, and I think last week or this week they just passed the bar of 250,000
Exactly.
species
yeah.
in the database.
Really?
Yeah.
Yes.
So
yes.
250,000
250,000.
marine animals now registered in the World Register of Marine Species.
Yes.
So that's pretty much the same number of plants on our planet. So it's a lot.
Wow.
Yes. You're an editor?
Sorry?
Are you an editor on worms?
I am an editor. Are you an editor?
Yes, I am. So that's why we know this, because we got this newsletter.
Yeah.
We're celebrating. It is a fantastic page.
Mm. So those pages are
Good.
maintained and curated by people like us, experts in
Experts for their group,
Yeah.
and I think they're worth gold, especially when you're looking up names that have changed over time and things get moved, like to another genus when more species have been found. Then you can take track of that, and you also see what other groups have been described.
I think this is Sacocalix.
I will enter.
Can we collect that one?
Sorry, can we sit down here with
Or
the
is it
rough?
too far?
Or is it too...
We can try, yeah.
Then,
So I've been looking for that sponge since
Do
the
you
beginning.
yes.
Want to slurp?
No,
Just to
it will be
know?
destroyed in 10,000 pieces. So
Can we use the shovel?
Yeah, can use the
so
shovel.
it is very light
Where is it going to go?
and very fragile. There is an empty box on either side, so whatever suits you better.
Port side of the shovel.
Okay.
What's that shovel?
Port side. 1, 2, 3 or 4.
So it's probably connected to the rock
Yeah.
by a stalk.
Yeah.
So if with the
If it floats.
if you
Yeah.
can break the stalk, then I think you can get it. But it's going to be very light, so that's why it might be a bit tricky.
Might be attached to the rock, so you might
Bridge,
get the
can I come
entire
to a
rock.
stop there, please?
Yeah, but that's a big rock.
Yeah, I can't.
We don't have to.
I
Can
can't try.
can we do a close up first?
Yeah.
And
Sorry? Yeah, it's 50.
Just move it to center first, and then I'll do. Thank you. Oop, now the rock is moving.
And it's
we're moving away.
We're moving away.
Sorry.
No worries.
Just trying to look at the umbilical.
yeah.
These rhinites are living a feast.
It's a little bit higher.
Yeah,
here.
that one. Okay.
So as you can see, that's a very steep slope, and
Yeah.
you have to be a really good driver to try to settle in this on this slope. Let's see if there's some of your animals in there. Why not? Maybe there's something inside there, right?
Yes, there is, but that's why it's so fantastic to get them fully intact.
I can center it a bit more.
Okay.
Like this.
Yes. Put a little shrimp on top.
Yeah,
Isn't
I see that.
pretty?
But there's 1, 2, maybe 3 other things there, no?
I see them. I do see them.
Yeah.
We can't focus that any further. So
No.
I can try the manual. Sorry, it might be out of focus now for a little while.
We're trying to
Nah.
focus on the.
Not getting this.
No. Okay.
Well, come on.
Oh, nice.
I'm trying.
Yeah.
Yeah.
So you can see little crustaceans in the sponge, either for shelter or to eat it. And I've been looking for that sponge because, again, we think we know the genus, but we don't have a species name for it. It's a glass sponge. Glass sponges are usually very thin, so very fragile.
I wish to
Could
see
we have the
this.
lasers just to have an idea of the size? Perfect, thank you. So
Okay.
you can see that sponge is about 20 centimeters large.
Anna, do you see your amphibian?
Yes, of course.
This one?
Yes.
You know, there was no reaction, so I was, I had to check in.
But
on
I
you
but I
if,
silently moved the camera over so to get the zoom.
Okay, I'm gonna leave.
Hmm? Hmm? Look at this pretty creature. Come on, go to Paco's sponge and just sit on it while we sample. He's hiding.
yeah.
He knows what you're up to.
Are you all zoomed in?
Yeah.
Almost.
Wow.
Oh, yeah.
So the little bright lights that you see is the eye, right?
Yes, yes, it's crawling to the left and has its long antenna moved forward.
Do they have one eye or two eyes or
Depe-
complex eyes?
Depending on the group, this one has two, one
Okay.
on both sides. I assume it's the family Calliopeida. They're quite common in this area. And Anne-Helene and I have described some, also from this area, also sitting on sponges.
Mm-hmm.
So, if this is 20 centimeters, where do you want the shovel to come from? From the right?
I don't know.
This will be tricky.
The shovel will come from your right arm.
Yeah.
Yeah. Then Then you will place it in one of the boxes on this side as well.
What I'm going to have to do
Or-
first is take the shovel out and then re-grip it because it's facing the wrong way.
Ah, okay. Okay.
Are you ready?
Yeah, I'm ready.
I'm ready as well.
Yeah, we're both ready.
Bring the basket in.
How shall I call the sponge?
Saccocalyx. SACOLYX.
the old pictures.
Sampling.
Otherwise,
And it's now 30.
So the robot is trying to catch the scoop, and for that it needs to be in the right position to grab it. So that's the first thing they would do. Are these chocolates for us?
Yes, for everyone.
I actually think they're further off pilots.
You know, I have half one left.
We're eating here constantly.
Excuse
Are
me.
these
I was-
German cookies?
I was feeding the pilots with some chocolate pieces as well, not only
Yes.
myself.
Do you want to share this? Do you want a piece? Or
It's let them eat, but not
No,
during
no.
their operation.
Yeah,
Now
exactly,
They,
I
now
don't want
they-
to
now they
disturb them.
really have to concentrate.
oh no! Who is moving around?
can you zoom in a little bit?
Yes.
Who's going for the sponge?
Yeah.
This one.
That's actually
Show on that screen which one it is.
Yeah, yeah, I've shown them.
Yes.
This one, right?
Yeah. Exactly,
Yes.
yeah.
Yes.
Yeah. So you can imagine how difficult it is to subsample a deep sea organism.
Well, 2.5 kilometers above.
Oh,
Oh,
wow.
yeah.
That's it.
You're
Oh,
a
magic.
chief.
Wow.
Perfect.
That was
Yeah.
picture book. Sampling.
So,
Tell which one.
Okay.
yeah. So it's going to be box 8. So that's on your right bottom side.
Which one?
This one?
This
This
one.
one.
Yeah. It's currently empty, and that will be cast 13.
13.
No.
Oh, we lost it. We lost it.
Mm.
Can you see another one just while we're sitting?
No. Was it taken up by current?
Yes.
Yeah.
Yes.
Sometimes.
Yeah.
Sometimes we find things on the rock when we
Wanna
sample that. Is there
look
another
for
one?
something?
We will try to find another one.
Zoom in on the
Right now I
jellyfish.
don't see any more. Yeah, that's the challenge of collecting in the deep sea.
Ah, it looked so good.
It's not like going into a forest and picking up plants. It's a bit more work.
That was a jellyfish.
You want to put the shovel in?
But wherever this sponge will end up, it will regrow, like it will settle again.
It might
Yeah.
resettle somewhere else.
Okay.
Sponges can heal very rapidly and very efficiently.
Also, if you take a piece of a large sponge, it will regrow.
Yeah, absolutely.
Actually, like a coral branch, it also regrows, like an apple tree.
Can you zoom in up? Thanks.
Almost. Almost. I'm just zooming in while we're sitting.
Okay. We will continue.
It's doing it again. Oh no, it's the left.
Wow.
There are lots of these jellyfish.
Yeah.
You should learn
There's-
the species.
There's two.
Yeah, there are many. Really many.
Okay.
You can swap afterwards. Yeah.
Very good.
Wow.
This is incredible.
Yeah.
Do you want anything?
It's like a painting.
Yeah.
Rorschach from Dryland, can you please put out the sex on for a while?
Yeah, okay.
Okay.
So we are deploying our fisheye camera to get a nice shot at this cliff, this amazing cliff.
It's interesting how the cryonics are all on
Mhm.
one side, on the feather stars.
Could you try to stay on
And
the
on
that
the right
side?
side, as well as the filter. More filter feeding is happening clearly on that side.
Oh, that's amazing! Look at
Yeah.
that.
It is a steep cliff.
Okay.
Another one.
Camera
I think that's
Camera
again
what?
my species.
Oh,
Well,
wow.
could it gone on on a flatter place, please?
Yeah, yeah, yeah.
It's
Yeah,
easier
yeah.
to stop.
No, but it's good. It means it's around. So
Oh,
we
these jellyfish.
yeah. And all these feather
Can we
stars.
continue the move, please?
Is the camera out?
Yes, it's there. So we need to take it in next time we collect. We shouldn't forget. Wow.
Can we turn on the lasers, please?
Yeah, on.
Thank you.
Apparently the feather stars love it. They prefer that side.
Oh, look how many jellyfish.
Yeah.
1, 2, 3, 4, 5, 6.
Amazing.
So how deep it is? 2,450 meters.
That is very deep.
Okay. Do you know how many species of amphipods are described every year on average?
No, I don't.
No?
There have been a lot of discussion lately about cryptic species. So, to separate species from each other, when to call it a new species. Because especially a lot of the groundwater ones look extremely alike, but they have a genetic distance. So they differ in molecular terms. But that's very tricky to describe.
Two different species of jellyfish.
And an anemone?
So there's the red one, and then there's one that's
There
more
was-
transparent, or
Yeah, there was
Yeah.
another species.
this one maybe, or I don't know. I think we saw another one that was transparent, more transparent.
Yes.
Oh, we can zoom in on it. Oh yes, finally.
Don't yet before the zoom.
Finally.
It makes it so wobbly when
They're really
Zoom.
active.
yes, very pretty.
Mm-hmm. So the thing standing out now on the camera is the fisheye camera, in case some of you are wondering. And it's extremely fragile, so we don't when we don't need it, we try to pull it back inside the robot. But when it's scenery like this, it's really nice to have this 180 degrees view of the landscape.
We have a lot of monitors here, but we cannot see what this camera is filming.
Ooh, fish!
Slightly.
And
That's an amphipod, right?
yes,
Is there a common name for amphipods? Like a general term, non-scientific term?
yes, there are in different languages. Is I think beach hopper
Beach hopper.
is the English term.
Sometimes I mistake a crinoid for a sea spider,
Mm.
and I just get all excited until
Me
I
too.
realize
Me too. I do exactly the same.
it's
After we've seen so many sea spiders, they've got a similar color, a similar size, and they've also got lots of arms and lots of legs that are bent just before the middle part.
exactly.
I had it a couple of times now that I had that mistake.
To be honest, this morning when I woke up and started to watch the video footage,
Paco,
I'm trying to follow the jellyfish.
they'll come to us.
Well, like this, you've spotted the huge lights on top of the ROV, without which we wouldn't see anything
there was
So-
the second species. Yeah.
of
Of
jellyfish.
Yes.
Mm.
You asked for the name of the amphipods. Our Norwegian colleague Anne-Helene just told me in Norwegian they're called hoppekrebs. I don't know how to pronounce it, but hoppe, I like that.
So a little bit like beach hoppers, but
Yes. But I think the beach, well, I don't like the beach as much as the hoppek.
mm.
And in German it's a Flohkrebs. Look at this one to the right.
Mhm.
What's that? Can we zoom in on them? What's this?
So there's a woolly sponge, a pompon sponge.
Stop it.
Lots of feather stars.
Escape.
You can see here that it's quite steep in the distance. Mm. I think sponges like the company.
And the feather stars.
Yeah.
Just entertaining them. Well, you may think so. I don't. They might be really annoyed that they don't get enough so much food anymore if they're covered by feather
Maybe,
stars.
but maybe
you
the
know,
feather
and
stars
not-
clean them as well. I don't know.
You wish they eat for themselves.
Mm.
Paco,
Yeah.
on your previous stop, I saw very small stock sponges. So if you happen to find a small stone or on your next stop, and there are small sponges like this, it can be very small.
Yeah, I saw
Maybe
them.
it will make it to the surface,
Yeah.
and that would be
Yeah.
wonderful for the picture.
Yeah. Yeah.
Not so big, very small things.
Yeah.
Small stock sponges.
We will try.
Nee.
Okay.
Verstanden.
Keep up the good work. That's amazing.
Look at this one.
Thanks.
Look at this one. It's covered as well.
Astrema mega antifolia.
Mhm. Yeah, that's good. Enjoy. So let's find a little rock.
It wasn't a joke. It was actually finding a sample suitable
Yeah.
rock size.
So we have a lot of photographers on board, and they want to make nice pictures of these animals. So they also have requests for some species. And it's very steep and
It's
very hard
It's
rock.
like being in front of a wall,
Yeah.
going up.
So it's pretty much impossible to sample here because you can't get a grip on much.
Just climbed 30 meters because it's now 2,418.
Yeah. tulip sponge.
See the fish?
Oh no, I didn't see it. Oh, there it is again. I don't know if it's the same fish.
I only locked it once. Will this camera stay out?
No, as soon as we will collect something, we need to bring it in.
When we collect something? No.
Yeah.
Okay.
But I think right now, since we can't really collect on the cliff, we just want a nice panoramic view.
It's quite large.
Yeah.
Looks like a potato.
Yeah.
Yeah, so we just started.
Asconema mega atrialia. This is a glass sponge. So for instance, something like this, you collect or it's too complicated?
I would have to have some thrusts.
So what you're going to have is you're going to have the ROV right up against there, but the arm has only got that much space to move.
Yeah, okay.
I think in the interest of this precious camera, we don't want to do that. That's why I'm asking if we can pull it back in, because if we get this kind of
Oh,
sampling,
camera, yeah.
we can't do.
With the camera,
Like
Yes.
on the fly, it will scratch. It will be too close.
Yeah, no, absolutely.
We've moved the camper in first before we've done any arm work.
now it's a little bit more flat. So
Okay.
if you find something.
Yeah, then I'll try to find the one that got away.
Sorry about that.
Yeah, it might be that, but I'm not sure. Okay.
It looks like lace.
Yeah. So is there a lot of current right now?
Well, when we sit down, we put the thrusters on to suck the dirt into the thrusters to keep visibility. Because it was so light and it was moving in the scoop, it flew out and couldn't even shut the thrusters off, otherwise we'd have lost it completely.
Yeah.
So
Of course.
it's a balancing act.
Yeah.
But anything light like that probably would be best in the port drawer because we've got room to manoeuvre. So if you do get another sample like that, best to put it into the port drawer if you can.
But then with the arm?
Then the arm will get to it a lot better than right on itself.
Yeah.
As you go out further with the arm, you've got more dexterity. Closer in, you've got very little movement, so it's more tricky.
Okay.
Okay, thank you.
Here's another tulip glass sponge.
with another shrimp on top.
Mm.
The red one. The visibility is fantastic. Think of all these sponges, they feed on what's in the water column. So it's great that we can see so far. I think it was a feather star moving to the right.
Wow, lots of tulip sponges here.
I'll just zoom. What about this one on top?
Mm-hmm.
No.
Mm. Here it's maybe easier to sample because it seems you can take a rock. They're
Probably
detached.
will keep
But do you
the
see
sponge.
I'm trying to find
Yeah, of
something
course.
for I'm trying to find something for solvent.
For solvent?
Mm.
So not. I thought the focus now is on your sponge that got away.
Yeah. But I'm also looking in case we find something.
Together.
Yeah. Mhm. Oh wow! Look at that.
And up we go.
Whoa! Beautiful. Wow! Look at those
Feather stars on the
feather
right.
stars on the crest of the rock. It's beautiful. Yeah, it's probably on the ridge of that rock that the current is the fastest, maybe, and brings more food.
Just as we mentioned, the good visibility, it starts to get a bit murky.
Mm.
And that's why I don't think the feather stars have an association with the sponges, because they're also on the rock.
Mm. Yeah, it's true. They're just trying to get higher in the water column. Mm.
Zoom in again.
Do you think it's possible to collect one of these two if you set it here? Or it's also a bit too...
Or grab it, or
Yeah, to grab it, yeah. Because they're going to be also very light.
move the sextant in.
Oh, yes.
Dry lab is this one, right?
Dry lab ROV.
Right out here?
Yeah, we're now going to sort of try and collect one of these mushrooms, and so we're going to be sitting on the rock face, so we're going to pull the sextant in.
That's fine. Pulling the sextant, but never say mushroom above quantity.
Tulip.
A tulip is better than
I think I've just been told off.
So we're going to try to collect one of these tulip sponges. We haven't collected any since the beginning of our expedition, Deep Arctic expedition.
So you control
We think we know the species, Amphidiscella monai,
Yep.
but to my knowledge no one has really
it.
looked at the genetics.
Kind of steep. I don't know if you
So this would be a fantastic opportunity to get a fresh specimen for
Too steep.
Yeah.
genetics. Too steep.
It's too steep
Okay.
to land here. We'll just continue flying.
If I have to land the RV here, so see where the tulip would be.
We'll find another one, and that's better.
Well, we
Yeah.
can try. What do you think, Rob?
Dry lab, do you want the sextant out again?
No, we're setting now.
Oh, is that?
Yeah.
Okay, I thought it's too steep.
Just to see if we can.
Or these or the other one?
Yeah.
That one's probably doable.
Yeah?
And
First, we will like to zoom in.
Yeah.
photo shoot. And is it brittle?
No, but it is probably flexible and very fragile. So it's going to be very light, like the previous one.
Are we good to go? Then lasers off, please.
Okay.
And there should be a little stalk behind it.
Don't.
Okay.
Don't.
No.
We could bring the rock, maybe.
Yes, but it seems
To be
like a big rock.
yeah.
Let's enjoy that view for a moment. What's this? Hanging out.
It's a sponge, but I don't know what's coming out.
That's what I meant.
I think I've seen this. I have no idea. It's like a sponge growing inside another sponge.
But they are on several. Look down here, there's also one hanging out.
Oh,
I
yeah.
first thought it's a worm, but it's another sponge.
Show me.
I mean, it could be because I think that's one of the sponge producing mucus. Could it be mucus? It's a bit strange.
But it's on several. Look. We have three, but we don't quite know what it is.
Yeah. Do you want flashes?
I don't think so.
Thank you. Thank you. Yeah. Thank you.
Thank you.
Super pretty.
Everyone is seeing this eat.
Do you think this is one of your?
I don't know. It's so faint.
Mm.
It could well be, but
I think it could be
it could be, but it's so light that I
because it's
can't-
moving inside the tulip sponge.
Inside the tulip sponge is a critter, but we don't know what it is. But chances are high, yes. It is. Also, the eyes are on the right side, and it's moving in the direction.
Yeah.
And they move forward, so they always go in the direction of their head.
And this one?
Yep. Well spotted.
Mm-hmm.
And they're the similar size, so that's also another
Yeah.
indication. So are you going for that one?
Yes. Yes.
Just checking.
Yeah.
Okay.
The other ones are almost impossible to correct.
Okay.
You ready?
I'm ready.
Assembly.
Ready when you are.
So do you want to what? Do you want what?
She makes this difficult.
Yeah.
Right.
So let me just see my space.
Yeah. That should come out.
What's it cost?
That looks good.
13.
Thanks.
How much thrust have we got going for it?
10.
So you might be able to Bridge, can we come to an all stop there, please?
Oh, it's done.
Okay, so 7 is
That's it, yeah.
So since we're in sampling mode, we've asked the ship to stop and remain above us, above the robot. So normally just behind it, it should have a stalk. Ah, yeah. You see it. Perfect. Yes.
Yes, yes, yes.
And that should be rather solid. But it is thin.
shrink side by side.
Are we trying for the rock or the stem of the tulip?
This
Try
time.
for the rock, to be honest. That's,
I think the rock is going to be too big.
Yeah.
Or I'm not sure
Mm.
it's even detached.
It's attached to the nucleus of the Earth. Maybe this side. This top.
just tried that, but the rock stopped me from there.
Yeah, exactly.
Yeah.
Yeah. Yeah. Yeah.
That's good.
Yeah. Perfect. Perfect.
Okay, so now
That's right.
What is more convenient for you, right or left?
Yeah, at the moment,
The-
right. Is the left one, yeah.
Left one. Okay, so that's gonna be your upper left box.
The very first one to the left. Yeah. Oh, is there the map in
Yeah,
there?
there's a mug.
Yeah.
Okay. But is the
Can we-
mu- is there something?
Cup.
There shouldn't be anything in the mug
Most
yet.
the cup.
I don't think.
The coffee mug.
Yes.
No, but then
Put it on top.
Oh, right.
let's put it somewhere else
Can we
because
put
we
it on
don't
top
want to
of
mix
this
it
one?
with the sediments.
No, because if you...
Okay, can we put it this one?
Or this one, because they're. I mean,
any of the other
yeah,
two
any
on
of
the
these
right
two
side
would be
will be
good.
fine.
Okay.
Thank you. Just right mug.
Yeah.
We're talking about our coffee mug here. That's another
Can you
sampling
zoom
unit.
your camera,
Yep.
please?
Yep.
Yep, sorry.
Oh, I've got some other stuff with it as well.
Rhinoids.
That's fine.
Oh,
That, oh, and lots of critters inside. Look at it.
Can you pan down any more?
Yeah.
That'll do nicely. That's big.
uh-huh.
It is bigger than I thought.
Yeah.
Right, so I have to
I
go
wouldn't
back.
mind turn it upside down. Yeah.
Yeah.
So they.
Then maybe it's better in that.
Okay,
There's a
I'll-
bit more space.
Oh no, the kind of off. Yes, put it. If it doesn't fit there, then go to the coffee mug one.
No, but I think it should fit in there.
Anyone is actually fine. Will be.
Look, you see it fits quite well.
Yes.
Actually, here it's a bit.
We have a different. Well, we have different cameras, so it's not as easy to see from the main one.
Yeah, perfect.
Yeah, you're doing really well. Perfect sampling.
Yes.
It's the crime. Oh, there, I would have liked to go in. It's like flower picking. Ooh.
Perfect.
And they were.
Perfect.
Thanks a lot.
Nice.
So it's cast 13.
And 2,383 meters. Let's continue our flight. Thank you. We talked about our coffee mug. That is actually a sampling unit. It looks like a mug, and there's a net attached, but it's only suitable for soft bottom. Trying to scoop up some sediment. Not here.
Yes, you continue. Yep, continuing, yeah.
Some anemones. Zoom in a little bit on those. Oops. Here on our left, light pink.
Right here?
Yeah, just no, I'm just registering them, so just to say what I'm doing. And more feather stars. Shrimp.
So feather stars are the same group as sea stars, sea urchins, sea cucumbers. They all belong to the same group.
They fossilize really well. That's why it's possible to know.
The crinoids.
Yes.
Yeah. So sponges don't fossilize very well.
No, leave it around for pods. We have to give that point away.
Apart from one group of sponge called the rock sponges. They're super hard and they fossilize very well. And we can go way back in the Trias, Jurassic, Cretaceous, and we find lots of those rock sponges. So at the same times that dinosaurs ruled the terrestrial world, we can imagine that maybe sponges ruled one part of the underwater world during the Cretaceous and Jurassic.
Next to the feather stars.
Next to the feather stars. Probably some amphipods as well. No?
No. They took their time. Hello, Jenny.
Hello guys.
Do you want to swap?
Oh yes, please.
Okay. I'll swap with you now.
Yep. That sounds good.
Okay, good. Let's go. Enjoy.
Thank you. How's it going?
Good.
Good. How many?
Jenny has joined us.
Hello.
Maybe you can say where you come from or Which institute?
Yes, sure. My name is Jenny Neuhaus and I am from the Senckenberg Society for Nature Research in Germany. And I am based in Hamburg. And I have a background in deep sea research on benthic invertebrates, which are small animals that live associated with the seafloor. And we are staring at them like that's all benthic animals that we're currently seeing, and I cannot get enough of this. It's just amazing. We've already seen so many feather stars today, and I don't know, they just amaze me. They're so beautiful.
I mean, this view is extraordinary.
Yes, it really is. But I have to be honest, I don't think I've ever seen that many feather stars in one patch before. I know this is not rare, but to my eyes it is, because...
No, it's really beautiful.
Yeah.
Could we have the lasers back on, please? Thank you.
Would you mind stopping there for a sec?
Yeah.
I would really love to get a shot of that, of this little edge. May I?
Sure,
Is
absolutely.
that okay?
Yes. So we have the privilege in the Rock Shack that we can actually, the scientists can control the zoom and the direction of the camera. So we are the ones to blame if if you have zoom movements that are a bit too fast.
There are two species
No, I
of
see them.
jellyfish in the picture.
No, I see them.
Yes, and you see how different, they're really different.
Only 4 tentacles.
Oh,
that
there's
are the
another one. There's this third species maybe. Oh, okay, that one.
But could it be
quickly.
ctenophores? They're a bit too fast.
Yeah, they're very fast floaters. Little dance, thank you. Okay, now I wanted to zoom in on the feather stars again, because I just think they are so incredibly beautiful. Especially along this edge.
Mm-hmm.
Wow.
Great, Jordi. Can we continue the move, please?
Can you move 310 at point 1?
Thanks, Baz.
Amazing. Thank you for the stop. We may continue. So currently we have Andrea and Rob as pilots, as ROV pilots. And they're doing an amazing job flying the ROV in this environment.
Look at the background.
Wow.
It's just cliff after cliff.
Yes.
And all of them absolutely covered with life.
Yeah. Really amazing.
So I hope you will never think again that the deep sea is empty.
Yeah.
It's your choice if you want to go to the next rock or to the further one.
So either here or either there? Is that what you're saying?
Yeah.
I'd say port side.
Okay. Yeah.
Good.
Let's check out what's up. I've already logged the fish.
Oh, the change just left. Yeah.
What are we looking at?
Well, he's looking at us.
Hey, buddy.
Not easy to follow.
No. Okay. We've got a little glimpse. Actually, where's our species guide? Because I personally don't think I have seen that one. Now it's gone.
Yeah.
Alrighty. I have logged eel pout, but that could be wrong. In fact, you know, because I think it really resembled the sea snail, like a bit.
Mm-hmm.
Paraliparis.
Yeah, maybe it was this one.
Not sure. I haven't worked with fish ever, so it's.
Yeah, me neither.
Yeah.
That's a very steep wall,
Amazing.
completely vertical.
You can look in my camera.
That's the tulip again.
Yeah,
That's a different one from the lollipop sponges, right?
yeah,
So we call them tulip sponges. Okay.
yeah.
So for those that might just recently have joined this live stream, we're logging the species that we see.
And we're a little bit on the outskirts of Mohns treasure,
Yeah.
which is on the Arctic mid-Atlantic ridge, which is the border between two tectonic plates. The North American plate on one side and the Eurasian plate on the other side. This is where new oceanic crust is created, but very slowly. So this is what we call an ultra-slow ridge, meaning that only less than 2 centimeters of crust is created per year. So it's really not much. Not much volcanic activity here. The maximum speed you can find in the world will be in the Pacific, where it can be up to 14 centimeters per year that is created,
A little bit.
meaning that the tectonic plates will move and will spread apart much more rapidly than here. Here it's ultra slow.
Right. And we're currently at 2,357 meter steps.
So all the rocks you see are volcanic rocks, because of course there were a lot of volcanoes in this area that produced a lot of magma, and when the magma solidifies, it becomes a volcanic rock. And all of these black rocks that you're seeing with us are volcanic rocks. What we generalize as basalt, usually.
Oh my God, look at this high density of crinoids and sponges. I mean, this is incredible.
Crinoid city.
Yeah, really. Oh wow, I will log that.
Rhinolandia.
Oh.
Oh, the fish is following us.
So it's gonna appear if you look down.
Yeah, there it is. There we go. Oh, beautiful. Yeah.
Like Jenny said earlier.
Oh, it bumped into a sponge. Oh.
Maybe he's blind.
Yeah.
Oh, little guy. But at least it was soft, right?
Yeah.
Ooh.
I would call this one the clown nose. I don't know why it makes me think of a nose of a clown.
Let's try if we you know, we have some jelly experts that are really yeah, it's difficult. Sorry, guys.
It's challenging. Yeah.
Ah,
There's another one.
Oh,
Oh.
they're both of them.
And it's tumbling away.
And sorry. We'll try again. There's another one coming up. So this will be now a bit crazy camera movement here because...
And it's gone.
And it's gone again. Let's focus on the benthic sessile animals that are not moving away that quickly. I really can't get
You
enough
know
of
what we
this.
should keep our eyes open for? Storozoa. Because last time we saw them in this environment.
Yes, you're correct.
And we didn't collect, so...
Yeah, you're right. So there's this bell-shaped Staurozoa, they're called. It's cnidarians, like they're in the same group as jellyfish.
Yeah. Maybe we call them the stocked jellyfish.
Yeah.
Is that not a thing? I think so.
They are really pretty. They have these little pom-poms shaped.
Yeah, the tips of the tentacles. Yeah.
Exactly. It's hard to describe until you see one, and then it's really clear.
Yeah.
We can do a bit of zoom, like a gentle zoom to see if we spot anything.
So I change the ship's heading to 270 west. Yeah.
There's always this balance between zooming in and trying to spot. Oh, jellyfish! Look.
That's the other species you were talking about. Yeah.
What I want to say is that there's a balance between the zoom that we need to see smaller critters. Now there are plenty. And the movement of the ROV and the video in general that obviously increases the more we zoom. So but I haven't spotted any, even though when we zoomed in, I haven't seen any.
Me neither.
Okay. Let's give it another try here. For the jelly guys.
Oh, wow. It's keeping its Oh, perfect.
Yeah.
Oh, that was good. We had it in focus for 2 seconds. and then you saw those 4 tentacles up in the air.
Yeah. Look, there's the fish again. Over there to the right. It's hiding. It's lying flat on the slope.
Oh, I thought that was a rock. Okay.
I think it's a fish.
Yeah.
It looks soft to me.
Yeah, clean surface. Yeah, it's the fish. It is.
Okay.
That's the one.
Having a little break. Maybe it's the one that got hit. Like hit its head on the sponge. Yeah, well.
Do you have the water temperature here given?
No, you guys have it there.
On the CTD.
We do have the water temperature.
Let's have a reading on that.
If I am not mistaken, we have a minus 0.65 degrees Celsius.
Yeah. So negative temperatures right now at 2,300 meters
Yes.
depth.
Oh.
If you look down, you have
Oh.
fish.
Hey.
Beautiful. Boom.
Oh. Yeah. They are obviously not used to the light. So they get a bit dizzy from it, but they will recover. It's just for a short period of time that we are illuminating them and their habitat quite intensely, but
Yes.
that's just, we can't avoid this.
So usually everything is dark here. The light and the sunlight cannot reach these depths. So all these animals constantly live in the dark.
Yeah. I have to say, of all dives we've done so far. Since we arrived today, like since our shift started, this has been the most constant habitat, I would say.
Yeah, I mean we saw a very similar habitat on our previous seamount at the same depths.
Yeah.
It is very distinct and very. Oh, I think this is the one I wanted to.
Which one?
Do you think you could get it? It's this one.
Are we trying to sit?
Just see if we have.
Sure. So, this species is special to you?
Yeah, because I don't know the species. It's been reported. I've seen a couple of pictures, but it's not been identified to the species level.
Mm-mm.
So I'm curious.
Try, I don't know. Which one is it, Paco?
It's possibly that one, but I.
There,
Yeah,
there you go.
it's this one. So it must be. Yeah, it must. No.
It's to the left of the rock. It's that one.
That.
Yeah.
Bridge RV, can we pause, please?
All stop.
I'll just zoom out a bit so we see what's happening, so we can orientate. There's another specimen.
Are you?
Oh yeah.
In case we don't catch that one, we might get
Are you
that
going to
one.
grab or slurp? Are you going to grab or slurp?
Slurp, no, it will be destroyed.
Mashed.
How
Yeah.
about the scoop?
Scoop we tried, but they are so light
Yeah.
that you scoop them super well and then
Yeah.
Yeah.
they
Like
fly
what?
away.
The thrush just pulls them.
Yeah.
Are you?
So this sponge has about 20 centimeters in diameter, so
Huh?
what we see on the video often looks very large to us.
Yeah.
But when the specimens get on deck
Yeah.
and we have them in our buckets, we first see the real size of them. It's sometimes really hard to imagine how big they actually are. We might be able to just grab the stone.
Yeah, maybe grabbing the stone could be an option. I don't know if it's sitting on.
If
Yeah,
it's,
see where you go.
Yeah.
let's see. Let's have a
You
still.
can try.
Just give us a minute, Rob. We'll just have a little still.
I think that's a baby woolly sponge.
Mm.
There's more room to start.
Can you
Yeah.
Yeah, okay.
tell
Go for start.
which box were headed?
Oh yeah.
Which side would you prefer?
We're going to go to start. I think there's more.
So this one, yeah, bottom right is empty.
Could you please turn off the lasers if you have the chance? Thank you.
Oh, and there's a little shrimp on it.
Yes, it's a nice still image of your specimen. And we've got what I believe are anemones. We have some other sponge species. Which cast is this? 14?
14.
All right. So whenever we collect animals, we give the event, the sampling event, a number, so we can retrieve the information we need for depth and location and time afterwards when they're processed and analyzed.
Paco, will this break if we pick the all up?
It will break a little, but that's okay.
Something, yeah.
These rocks might
I can nudge it and see which one's on.
If you cannot grab the rock, try to grab it from the bottom.
Base.
It will break a little, but that's okay because the rest of the
I
sponge
use
will
when we
be.
go to let it go, that it doesn't float over.
Yeah, that's
we
the.
need it attached to something.
Ah, yeah.
I'm sorry, but I found a picnic on it. Look at it.
Oh yeah.
Just
Okay, so when you look, you have the sponge, and to the left of the sponge, let's say at about 8 o'clock, there is a sea spider that is very well camouflaged on the rock, and it has just the same coloration as the debris or like the sediment that is on the rock, but it is a sea spider. And I think I'm maxed out on the zoom, but
And during our live stream, was it yesterday? It was yesterday, where you saw a lot of pycnogonids.
yes, we did.
So if you want to rewatch that pycnogonid action, you have to go back to our previous live stream, where we saw actually adults carrying their babies, which was pretty amazing.
Yeah, that was incredible. Shall we?
I'm ready.
Okay, me too.
Yeah. So
Is it okay if
yeah, absolutely.
You're fine.
Yeah.
Okay.
The ROV needs for the sampling. We need 2 people. Andrea is maintaining the robot stable in the water column, while a pilot is using the arm to catch the sponge. So it's a 2-person's job, and it's very tricky, as you can imagine.
And then we have us steering the camera.
Yes. Jenny controlling the camera, and me being a bit nervous for the sponge.
Moral support all the time. And the decapod can leave now. It's alright. Escape while you can.
Ooh.
Yeah, it is very fragile.
I wasn't expecting that the rock would break so easily.
No, I wouldn't.
Wow.
Yeah, it is. It's very hard to predict sometimes what is going to happen when we just touch the sediments. When we just look, the decapod disappeared
Oh, yeah.
now. That's good. Bye.
Yeah, maybe this part is part of the whole rock.
That's sticking up, right?
Yeah.
Mm-mm.
Because then it would be really.
It now. If it's too difficult, it's too difficult.
Just clear a little bit. Yeah. We're gonna clear a little bit.
Sure. I mean, this is basically like in the mountains, right? You touch one stone and they all keep rolling, and then we have these sediments in the water, so they create a lot of
I think I
dust.
saw it fall. No?
It's here. See? So it's this one.
Uh-huh. So it's right. Okay, it didn't go too far. I think I see it. Yeah.
Just slid down a little. Now the little edge is gone. It was nice to grab it. Let's see if we manage. There we go. So now we've got a lump of clay in the manipulator. Yeah, we need to be patient. Very patient. Let it clear. Let it settle. Give it a new
Oh, you still see
go.
it? Oh, wow. Because we don't see anything on our screen. So yeah, you have to imagine that there's many different cameras around the robot, and we're just showing you the front view, but there's actually a lot of small cameras where you can see the bottom, the sides, and so on. So the drivers of the robots, they have much more control of the whole robot. I want to do this. You know?
Yeah, exactly. Turn on the fan and just get it out of our way. Hi, buddy.
So again, we're collecting this species because we don't know what species it is, and without collecting it, we cannot give it a name. But once we've given it a name and we see it again, then we'll be able to name it directly on the movie, on our observation. So
Without collecting it.
without collecting it. So
Yeah.
we need at least one individual.
Ah, it's clearing. Maybe not. It might be the clay lump that is in the claw still.
Mm.
So yeah, there's always lots of patience. Ask from everybody to retrieve these very, very precious samples. So you might now start to get an idea of how precious they are, given that we are at 2,341 meters depth and need, I don't know, lots of, yeah, energy, resources,
Logistics.
skills, everything needs to fall into place to be able to collect single specimens from the seafloor, to explore their diversity.
Okay.
We're giving it another try.
Yeah.
And you cannot see much at the moment, but the pilots have a bit of a better view on the specimen. It's a tricky one.
It's a very tricky one, yeah. You have to imagine we're on a cliff and we have this huge robot, 3 meters large. That's stabilized itself in the water column.
I mean,
Yeah, I think you have it.
oh my, we got it.
Yeah.
You have to do it template. I have to remove the
This one is totally empty. The other ones have lots of sponges already. No. And I'm afraid to put it there because there's already apparently other sponges, it says. I'm not sure what's in there.
So basically what's happening is that these drawers have been
Or you could actually put
pushed
it there.
out
I think right there there's
in
only
the front.
stone.
and then they have 4 compartments and
Yeah, but then do that.
we have to be very careful with where we put specimens to keep track on what we sampled in order to separate it again when it comes on deck.
No, I can't put the drawer out, so I showed her down.
It's going to be B7.
It's actually going on here at the moment.
No, look.
Ah, I see. Okay. I didn't want to confuse you, I'm sorry.
It's okay.
Sorry, Papa.
Just turn your thrusters off. Your thrusters off.
Try and get down a bit more pitch forward if we can.
At the minimum.
That's it.
Yes.
Ah, perfect.
Brilliant.
Thank you so much.
Just all the way and actually start between the jaw.
So that's going to be a very precious specimen. Thank you to
That's
the
off the forward thrust of it, Andrew, as it comes in.
to the robot drivers.
The arm's going to go on the
To our pilots. Yeah.
shoulder up more. As you start. As long as that is going to
Thank you guys.
Yeah, thank you so much.
This is a trailer.
Yeah. Are the lasers back on?
stay.
Thank you. We're ready whenever you are.
Just hold the arm.
Good job, Rob. That was very challenging with the bad visibility. And we continue our journey along this volcanic mound.
Bruce RV, can we continue with the move, please?
Move 310.1.
On your chin, not having anything on your stomach. If I turn this side.
Yeah, come back on your.
Leaning back there for a second. It's really. That was intense. I'm very nervous. Like because it's so precious and it's so easy to lose them in the current.
No, they're extremely precious specimens.
I think that deserves a piece of chocolate, actually.
Let's have a piece of chocolate to celebrate.
Yeah. There you go.
Thank you.
Anyone wants a piece of chocolate?
Oh, thanks.
Mm. So we're back on track, huh?
So I have another sponge expert that's watching us right now, and who's giving me advice on what these some of these sponges could be, because he's a glass sponge expert.
Oh.
Celso Domingos, if you hear me, thank you very much. You know him? No. He's from Brazil.
Nice to meet you, sir.
And he's doing his PhD on glass sponges from the Atlantic. So I always listen to his suggestions. He knows them better than me.
Did he have a guess already?
On several that we found.
Oh,
yeah,
wow.
Yeah.
That's really cool.
Mm.
I'm really eager to hear from our jellyfish experts about especially the red jellyfish that has been quite abundant in this area, but also the others.
Climbing. Not going forward.
Just checking because sometimes messages roll in on species. Is that a... yeah. I thought maybe we had something else in the water column, but it is a decapod. Oh! Stay with us!
Always a
Aw.
pleasure.
Yeah.
A pinch of color.
Very pink. It's very nice. So, probably many of you know this already, but lots of species in the deep sea have reddish colors because red is the first color that disappears with depth. You can already see it at diving depth, basically, when you dive at 30 meters and you bring a color chart down with you of the rainbow colors, you will see that the red color just fades away. You can't really recognize it anymore, which is the perfect camouflage in the deep sea. And although there's complete darkness, there will always be some species that can produce light or even see in this darkness. And so you gotta hide, or you will be eaten.
What is your favorite animal?
Oh,
There.
in this particular
I think I know the
environment?
answer,
Yeah,
but I'm
you
just
do. You do.
I'm just asking.
So it really depends on the habitat, but right here, right now, I have to go for the feather stars because they really amaze me.
Aha, then I was not expecting that answer. Okay.
I am full of surprises. No, but yeah, here is the feather stars. But like yesterday, it definitely were the sea spiders. It really depends. I have lots of favorites. It's very hard to tell sometimes. I also really love sea stars, and I am a bit sad that I missed the sea star from this morning. Feasting
Yeah,
on the
they're not at
feather
all-
star. No,
Not at all there.
they need more. The Hymenasta,
Yeah.
the specimen that we observed, they need more soft bottom. They thrive more in soft sedimented plains rather than rocky substrates.
So that makes me think also that we still have the mug to use. We haven't used it.
Right,
Yeah.
right.
So
Yeah.
you tell us when you think it's a good place, a good stretch of mud we could sample.
Yeah, I'm not sure.
And we have the cores, the 2 cores
Right.
still.
We still need to do some push coring.
Mm.
That's right. Thank you for reminding me. That is quite a good point there. Mm.
And what about scaphopods? Have you found any here when you
No.
were sorting?
No.
Why is that? They don't live in the deep sea?
They
or?
do, they do. There are plenty of scaphopods
Okay.
in the sediments. Maybe even more than we think. But you might need different gear.
Okay.
You know, you might need gear that takes more of a larger sediment sample to sample them. Although there are sometimes species that live more on top of the sediment than inside, or they can crawl or move on top of the sediment. And if we would encounter this, that would be extremely special. I don't think we will. You need to gently deploy a sled on a flat sedimented plain,
Mm-hmm.
where you then take a very small fraction of the seafloor on deck, and when you then sort through that sediment,
That's when you find them.
yes,
Okay.
that's when you find them.
Oh, look at that.
Oh, hey, oi, oh. It's like you see it and then it's already gone.
Mm.
Ah.
Maybe you can put it in the off-up. Fish.
Oh, yeah.
Fish to the camera.
Fish straight up. Still sponge grounds on, what do we call this? Say, yeah, let's keep the push course and the mugs in mind,
Mm,
but just maybe fly
because
a bit first.
Mm.
Yeah.
We're still just at the beginning of the
Yeah,
transect.
we should cover some track. It's a tulip again.
So the rough pilots are changing chairs. Of course, they cannot be driving and concentrating all the time. It's an intense job.
You can go first for dinner because you were flying last. Take time.
What's that? It's a Kirognas, I think. It was
Mhm.
moving like would be nice with a little cephalopod now, you know,
Yeah.
once in a while. They could drop by like this morning. And this dive has been going on for many hours, and during the morning hours, our team encountered a Dumbo octopus. That must have been a very special experience.
Yeah, I was sleeping, so
Yeah.
I didn't see it. You
Yeah.
were sleeping as well.
Yes.
Yeah.
There's always someone asleep on the ship. And there's always something happening on the ship too.
Mm.
Yeah. I would actually say that, I mean, there's still lots of sponges and they still cover the ground, but I would say maybe we have a bit less than we had previously.
Yeah,
Oh no, yeah.
maybe because we have a bit more sediment as well. So sponges don't like, some sponges don't like the sediment. They prefer to settle on a hard substrate, such as rocks or other sponges. So we're pretty much the second people maybe to film this area. The first one came to the same locality, but a bit outside of it. So we are still the first humans to see this place. But thanks to the first people that came there, we knew it would be worth it. So we already knew it would be quite rich in sponges and so on. And that's why we picked that area as well. So every time an expedition comes to these places, our knowledge increases a little bit.
And it's very interesting because we have been diving now since the very early morning, something like 6 a.m.
Yeah.
roughly.
Yeah.
And how many meters of the seafloor have we actually covered? Like maybe 1.5 kilometers, maybe.
Yeah.
So imagine that you need 10 hours to cover 1.5 kilometers.
So that's slower than if you needed to walk on that mountain. You would be much faster than us with a robot.
You could do this 3 times. Like it is, and of course we could go faster, but then we wouldn't see anything, and we wouldn't be able to identify anything. And speaking of which, I can actually zoom a bit in. The zoom is wonky again. You know, it's like sometimes it's zooming itself.
What are you raising this chair?
To the front first. Yeah. That's the front.
Yeah. Yeah. So again, patience.
Okay. Shoots forward.
Very few anemones now too. Like, and the crinoids are not that abundant anymore. Another jelly.
Oh, and this one is staying.
No. Are you kidding me? I was just about to, zoom in on him.
It saw you coming.
Or her. And gone it is. Gone it is. Askonema.
I will like that.
I could actually press the button, but I kind of like to type it because I memorize it more when I write the name more often.
No, it's just a shrimp.
So all the, maybe you would like to repeat that because I really love that name. The white sponges that are quite abundant here in the picture right now, what are they called?
So I call them the polar pompon sponges. But their scientific name is Lysodendrorix complicata. One of the researchers on board, Sergi Taboada, is planning a population genomic study on this species because it's pretty widespread. You find it at different depths, so it's an ideal candidate for this type of study to see how the different seamounts along the ridge are connected, And how are these also connected to further south in Iceland? So that's why we are collecting several specimens of that particular species, because when you do population studies, you need a good number of specimens to represent, to have an idea of the diversity within a species.
Polar pompongs. That is one of my favorite words for today. That's for sure. Either I haven't been in the shack when you mentioned this before, or I don't know. We've only called it Lysidendorix, but the polar pompon thing is new. That's so fun. And we have a beautiful Asconema.
Yeah.
And the crinoids are back, at least one. For those with trained eyes, I guess it could be spotted immediately. I will make a note of that. Now it's changing again. I have the feeling that now the rock is more. There's more rock.
There's more rock. That's the key, I think.
Yeah. Oh, beautiful tulip. It's not really push core area, is it?
Not really. Really on hard bottom here.
Yeah, the crinoids start to come back. Oi, what's this?
Oh.
The net?
Could we stop and watch this? This. We would like to do a close-up on this thing.
It's on you?
Yep. Here?
That one. What's that? The zoom is a bit. It's going. Okay. What is this? Oh, this is
Could these
pretty.
be eggs? You know?
It could.
Chains of eggs.
Could. I first thought it might be like some sponge because that could be possible.
No.
Wow, this is beautiful. The crinoid lifting its arm. Okay, so we have.
And I think all the knobs on the sides is Tentorium semisuberites, which doesn't look like that at all when we bring it to the surface. Of course, here they are fully developed, pumping, feeding in the water column. So all of these are Tentorium.
Right.
Here.
Beautiful. Amphipod.
Amphipod.
Yippee!
Yeah.
Let's just get it a bit back into the picture. The zoom is a bit doing its own thing again. It's a bit. Yeah. Yeah, it's.
Please turn it off.
Yeah.
Thank you. We can continue. Yeah.
I don't know. The interesting structure. Maybe some.
Okay, go Jenny.
You. Maybe someone knows. Eggs is always a good guess. It's.
Yeah. Okay, Bridge. We resume the move, please.
Okay, resume move.
But it's good we got this on tape.
Yeah.
Speaking of which, change tape in 5 minutes.
Yep.
It's good timing. And they want us to communicate via the comms to the dry lab that there's a tape change. You can just address it as dry lab.
You mean with the comment here? Okay.
Because they're not always listening to
Yeah, to what
us.
we're saying.
Yeah. Okay, no,
Yeah.
it's fine. I don't know.
This thing you mean?
Yeah,
Yeah, it's a bit strange.
it is. Yeah, no, well, it's alright.
Oh, that's a big rock.
Yeah, it really is. Wow. So we are very slowly but steadily ascending actually. So now we are at 2,295 meter steps. I think when we started our shift we were at around 2,400, something like that.
Yeah, it's just like climbing a huge mountain
Yeah.
underwater, though.
Is that a sponge too?
I think it's Asconema. It's just a
Yeah,
bit
okay.
cleaner
Yeah.
colored.
Oi!
Oh!
Hey guys! Oh! Oh, they will attach again somewhere.
Ah, but a lot of these big sponges. It's really good we collected one because now I'm
Yeah.
I'm not sure we have the right identification for it. Maybe
So you collected one of these large
tulip sponges.
bell-shaped.
Yeah, we did.
Beautiful.
Look at that. The overhang.
Yes. Really beautiful. Wow.
So is this your first time in the Arctic Ocean?
With an ROV, yes. That's definitely yes. But I have been sampling in the Arctic before. It's 8 years ago. We were on board the Helmar Hansen research vessel north of Svalbard, and that was very special to see this high Arctic fauna.
Yeah, that's very high. That's
Yeah,
probably higher than what we are now. We're at 73 degrees north.
yeah. We were at I think it was definitely 79 degrees.
Okay.
I think maybe one station was 80
Yeah.
degrees north.
Wow, that's very high up north.
Yeah, very much. Okay, we have a tape change in 1 minute.
Okay.
I mean, we could do it just now, couldn't we?
Yeah, okay, so 3, 2, 1, stop.
Rob, could you press the comms? Is that possible? Or
Dry Lab ROV, that's a tape change for the Kite Pro.
thank you.
So I did all that without moving.
Jelly.
Okay, and recording 3, 2, 1, recording.
All right.
Perfect.
There you go, Paco.
Thank you. I will write it down as well.
Yes.
So
Um,
UTC time.
16, 17.
So we are of course recording everything that you are seeing today.
Just as this beautiful. You may continue. I'm
Yeah.
just like,
Oh, are
Okay.
these stocked
yes. Osa.
there?
Yes.
They're really difficult to see.
Could we stop here for a bit, please? Because actually this would be
Reach
nice
over, we all stop.
for a push core.
All stop.
Yeah? Yeah?
What do you think, guys? Could we sit and do some push cores?
Not a chance.
Not a chance. Okay, alrighty. No worries.
It's all rock.
Yeah. All
Uh,
good. All good.
Let's move off again.
Oh, wait.
No, let's have a zoom in
Yeah.
on that rock.
Okay.
Do you think we could collect this? I think,
How? Also, can we pick it?
yeah, pick it.
So maybe you can do that. Or you start with it.
Because I think that's a species we haven't collected yet, and it's a sponge for sure. And Celso also, following us, told me this was a glass sponge, and I've never seen it before. So here we have a nice opportunity.
Are you talking about the ball shaped
No.
or the other one?
The other one
Okay.
that looks a bit, I don't know what to say.
Yeah.
It's it has a brown ring, and in the middle it's grayish,
Okay.
whitish.
Okay. I see. How would you collect it with the manipulator?
Yeah, with the manipulator, it's probably because, I mean, it's sitting directly on the rock.
Mm. Could we turn off the lasers for a second there, please?
Off.
Cheers. What do you guys think?
Only way would be to come in parallel to the rock and just pick it. But will it damage it?
Yeah, it will damage it a little bit, probably, and it's probably fragile. But
Alright, so start
I think this might be our best chance.
Can we slurp it?
We can try to slurp it. Okay.
Do we have any free?
Yeah, they're very free.
Uh-huh.
So I know that I shouldn't put anything on top of the
No,
amphipods.
no.
This one is a crust on the rock, so here we could put maybe something, but in those 2 and 3 we have amphipods.
Exactly, so it could be one.
1, 4, or 5.
Your call. But don't you think grabbing is better than slurping? Wouldn't it fall into...
I'm afraid it's super light, like the other, and that if we let it go just a little bit, it's just gonna... While with a slurp, it might be a bit damaged, but for sure everything will go in the box.
Okay, your call.
Yeah, let's try the
Chamber
slurp.
4, chamber 5.
Even 1, there's already
Fine for one, so-
there's a rock, but we can put this on top of it.
Okay.
I think I've just found an Ascidian.
Do you want the chocolate? Huh? Do you want the chocolate?
Or a baby Asconet.
Chocolate.
Or actually maybe it's the same thing. Maybe this one is a bit bigger and has become transparent. I don't know.
It is...
Could this be a...
oh,
a
sorry,
Asconet?
it's not me. It's the Zoom.
Oh, yeah.
Sorry. It's... Sometimes our Zoom has its own life.
Sorry.
Could
you... I can't see the...
Yeah, sure.
coordinates. You're a bit
No
closer
worries.
to the screen.
Yes, it's...
Yeah.
7, 10...
It's easy to get uphill.
So reaching up high for this, guys. We just got to see if we can actually physically reach it.
Sure.
There you go.
And if you take the little ball next to it as well,
If we can, yeah.
if you can.
Am I free to operate?
Yes, please. Go ahead. We might be too far away. I think we are.
That's the bit that comes up.
I think it's too far. So here comes the slurp.
So it's basically like a vacuum cleaner, and it will specifically.
Zoom in there, guys, in HD.
Yeah. Is that alright?
Yeah.
We're a little bit far out, but yeah. Ready? On.
Almost. Yeah.
Yeah.
Amazing. Wow.
Keep it on, keep it on, keep it on. Or off.
So it's Craniella.
Possibly,
Off.
possibly.
Off.
It could also be something else. So
Okay.
Craniella is usually a bit orange. This one is more white. So
Turn off.
Yeah, it's off.
So we are currently collecting two different species together because they
On.
grow together.
I think it's stuck against the rock. Yeah,
Yeah.
yeah, yeah,
Ah,
yeah.
there we go.
There you go.
Keep it on, keep it on,
Perfect.
keep it on. Off.
And off. Well
Amazing.
done.
Thank you so much.
Fantastic.
It's
So this,
it's ecologically important to collect them together, or it could, because they were growing together. We're good.
good.
So I will request. Could you request the push course again? Maybe we can do a push corner.
Do you think that if we do a push core here, there's not enough?
Send it.
No.
So it's all wrong.
Okay.
Okay.
Yeah.
Let's just take a time to zoom in on the tulip before we go, huh?
Sure.
Okay.
Beautiful.
It's zooming a bit. I need to turn off the zoom afterwards. It's a bit off. Yeah, it's actually. Yeah.
That's good. That's right.
Is that a sea urchin? No, it's also a sponge.
Rotate-
Yeah.
Rotate the shoulder. Forearm.
Yeah, it's the better boost.
Yeah.
That is very
It's
good.
very picturesque.
Yes, it is. It is.
Look at that with the little, even with the dust, I think it has a nice,
It's a sparkle.
yeah, nice touch.
Yeah. All right. I think we're good to go, aren't we?
Yeah.
After one
absolutely.
little shot here. Beautiful. We may turn the lasers back on.
On.
And we're good to go.
Yeah.
Okay.
Yep.
Okay, Rob, you get going. I'll get the rest of that. Yeah, it's just the way. Okay, bridge.
doing the move, please. Dumbo.
Yeah, you must be right that that was a baby Asconema. Because what Anne Helena mentioned was that we haven't seen any tunicates, any Ascidians during this dive, which is interesting because we had so many of them previously at other sites that we visited. So
Especially in the bamboo coral
they were everywhere.
field, they were everywhere.
In fact, we sampled one very pretty specimen. Look. Yeah, I will just keep it steady now. Oh. Hi.
As if we had known.
Hello.
Where are we? You're in transit 3 still, right?
Yes.
Yeah.
So still early on.
We are snailing ourselves up.
Okay, good.
And we still have quite a lot of space in the boxes.
Oh, you do?
Yeah.
Okay.
So we can continue.
Depends on your
Yeah,
plan.
that's why we're here. We're
Yeah.
trying to make a
Yeah.
schedule now
Yeah.
for having a time set for ROV to come back on deck.
Mm-hmm.
And in theory, just a rough calculation that we did would be that you guys would have another 2.5 hours until 8 o'clock.
Okay.
Sure.
That sounds.
Although we said 8 o'clock on deck, that would be.
No, I didn't. Well, no.
So, to any
End
of
of-
our
no, end of-
fish experts, what are we looking at?
I mean,
Till 8?
Yeah, that would be.
Sure.
I'm sure many would live it pushed out there longer. But okay.
I mean, we can.
Fairly. To prepare, once she comes
I
up
think I
on
will
deck
write
and you've had,
your
you've got the
parts
samples
still,
cleaned up
Yeah.
and
But
all of that.
I am tempted
How much time
to.
do you comfortably think you may need? I know you won't know 4 to 5.
This picture is a bit
4 to 5
bad,
hours.
so
So
we
if you
can't
had
really
6
tell.
hours minimum, then that would be pretty
Difficult.
Yeah, of course that's repetitive.
If anyone
Yeah.
live has any ideas
So
on
we
the
all
fish
get
species,
ours.
we appreciate comments.
That works out pretty much right because we've got to do
transport. So
Rough shack, dry lad, your sound is back.
Lovely.
Cheers.
So now after sampling with 2 push cores and the coffee mug for the sediment, we will now try and fire the Niskin bottles to get water samples.
Turn on over the loop until we get back to deck.
And while we're doing that, we'll observe our little crustacean.
Did we just have a little high five moment there because the Niskin
nobody saw that.
bomb
Nobody saw
and
that.
high five?
Nobody saw that. Nobody saw that.
Okay, okay.
What happened?
Niskins are closed. That's what it seems.
What happens in the shack stays in the
Is
shack.
this both Niskins or one? Perfect.
Okay, okay. Back in the game.
Can you also note
Okay,
that?
let's listen.
Yes, I have I have logged it.
Thanks a lot.
Sorry, I just took the little chance to, you know,
Yeah,
say
I know,
goodbye to our
but-
little. Okay, so we are now carrying very precious water samples from 2,183 meters depth that are about to be analyzed in the lab by our colleague Sergi Tabuada.
Just be little laterals ahead and lateral. So as you come up,
And this is great, great news.
there's your ridge.
Because the Niskins weren't always closing so well because of some issues that are
Oh, look at that.
now resolved.
Oh.
Oh my god.
Now that was
Oi. Ah,
that looked really good.
yes, it did. And now we've got them water samples.
Yeah, I'm really happy.
Yes.
Thanks for fixing that. They can't only fly this very well, they also fix things all
Yeah,
the time.
all the time.
Magicians.
Really. Yeah, no,
Improving.
seriously. Amazing. So we are now on a heavily sedimented slope. You know, sponge-wise, it was a very good timing that you came in and Paco left.
Enough sponges for today.
I
now.
have to
There are
say.
never enough sponges, I know. Well, I wouldn't mind another community coming in. Yeah, look at all the spores.
Yeah.
So many.
Yes.
They look like little dark stones from the top. When you zoom in, you can see that the actual holes
Yeah.
in the sediment. Look at that.
Quite a long, again, 0.2, okay.
Right, Jorah, maybe increase to 0.2, please.
So we
Can
are
you say
back
sponge
to.
community stopped? Can you
Oh,
just log
yes,
that, please?
definitely.
So no more large sponges.
Sorry.
No, I just was looking.
Yeah.
Another shrimp.
So we have sediment TV back on. Okay. Crinoid? No, maybe. Could be. I'm not. So we have burrows. Lots of burrows. Burrows. Yeah, that's a crinoid. I think these are stork crinoids. Sabelids. think it's sepalid tubes. Yeah.
I'm not entirely sure. You can see it on.
There you see it.
Yes.
Yeah. You see the. So I just said sepalid tubes. I'm referring to sometimes you see these branches, branch-like structures sticking up from the sediment, or like tube-like structures. That's a better word. And inside these tubes, they're inhabited by, and they are built by worms, bristle worms, and they have a very pretty feather-like sort of umbrella that they open into the water column to feed on on whatever is drifting by, to catch prey. And then whenever you come close, they just retract into their tube to be safe from predators.
Now the sponges moving.
Yeah, you're right. Sponges. And as soon...
Does it say sponge?
Yes, I will.
There's feather stars again.
Yeah.
Lots of feather stars are really taking advantage. There's one higher.
2 sponge specimens.
With at least 15 feather stars.
For a moment there I thought that was a cephalopod, but it was not. Ah, there's some rocks coming.
I'd like to stop in front of the rock, please. Is that still a good place to stop? Just have another zoom
Sure.
onto the rock.
Yeah, that's a good idea. I actually found one pycnogonid whilst you were not here. I had a little moment there together with Paco. Little pycno moment.
Look at that.
Oh, they're everywhere.
Mm.
Decapod.
Okay.
Decapods.
Look at these. I like
Salvage.
those. Looking overhanging.
Yeah, these overhangs. My God.
Oh, no. Look at all the different sponges. Oh, look at this! Look at this tulip sponge.
Oh my
they're
God.
hanging in there. Look. And, sorry.
Oi.
Look how the
How it's.
the decapod is underneath. Oops, and the amphibian and the feather stars on top.
Oh, yeah, this is beautiful.
Well, thanks for letting us zoom.
Let's have the lasers off there for a sec.
Thank you.
Wow. So this is, again, it's so diverse. Like you just saw previously, we have a huge sedimented area and there appears to be nothing, and again, that's not true. And then you have one rock and boom, they're all back. They just need some hard substrates to settle and they're all back immediately. It's incredible.
Look at the amphipod in the sponge. The lower left.
Yeah. What's that?
And there is what is on the crinoid, or no, on the sponge stalk? There's an anemone.
No, it's another crinoid.
It's another baby one.
Yeah, it's a small crinoid.
And then there's a crustacean critter on the crinoid that is almost translucent, that I just saw because it was moving. And then there's an amphipod in one of the tulip sponges, and then there are larger red decapods or shrimps outside of the tulip sponges. There's so much going on. You know what is missing?
Of course.
I mean, stating the obvious.
Bring up here, I'll go a little bit to the left.
Oh, wow.
I'll go up again.
Paco would now say, look at all the geodias. I will have to log in though.
Yes.
Can I stay much longer, guys?
No, not much longer. Okay.
Let's move.
Okay.
Let's move.
Okay.
Thank you.
You're ready. Thanks a lot.
Oi.
Tilt back down with your HD camera, guys. You're looking up at the sky.
Yeah, yep. That is beautiful sometimes.
The sponge is covered.
Yeah, it's
So
amazing.
stunning.
It's just amazing. Lasers back on, please. Cheers. Feather. No, fan shaped is what I wanted. Because now they're back to the fan shaped ones.
Yes.
All back. Back to.
Yes, the rocks are back as well.
We're still climbing.
Yep.
We are at 2,000.
2161.
2161. Correct.
So we've come up just over 12 meters since we fired an instance.
Steep climb.
So, I've just heard our pilot say 12 meters since we had the Niskin bottles and the coffee mug.
Yeah.
It's
Very
a lot.
happy about our coffee mug. Like, really.
The mesh size of the coffee mug is 1 millimeter, and that's exactly the size where we'll collect the critters that we're interested in.
It's dangling with its arm.
It's dinner time.
It is indeed dinner
Well, I
time.
was talking for the feather stuff.
Yeah.
Yep.
Right. Amphipod. We need to get another sheet. To go.
starting a new sheet here. Maybe we don't need it, but was that a pickup?
No, maybe it was a feather star. I'm not entirely sure.
I see.
Wait.
But look, some of them have so. You know, that one might have come on as well. We're too far away and we're flying too fast.
Something like
You
that.
don't want to keep going.
That's alright.
Yeah.
Where is it? Keep the image. Come on that way. That's the tip gun number 2. Number 1 over Andrews. Alright? Yeah. We're climbing all the time. Just go constantly thinking anything but it.
Yeah.
Seeing how it's all is.
In there. Huh?
Okay.
Wasn't even after. I didn't see much of the time.
You
No need for it. Some left. Be in the fridge, I reckon.
Now, back to normal, huh? Back to rocks, back to crinoids, geodias, fan-shaped sponges.
That was pretty.
Hmm?
The coral garden, or whatever it was, on the cliff face with all the colours.
Yes.
Saw it in the TV lounge, and it was better colour than what we've got here. It was really good.
Yeah, that was really pretty.
Okay. How are we doing for sampling boxes?
We still have one empty one on starboard side, number 8.
Okay.
But currently I see no need.
Me neither.
I'm very good.
Leaving that one. Yeah.
Exactly.
Me too. I just wanted to know.
Yeah, yeah.
So it was us. So it was.
It was us. It wasn't you. It was us. It was us anyway.
It's very unlikely that we are, or like it's very unlike us to
Look
do
at the
that.
look at the feather
And
start
we're
swimming.
back. And we're back.
Let
Nice.
it zoom. Let it just fly.
I want to look at it longer. No.
Longer is fine with them.
No.
We had that topic before. Yes. Someone touch my camera.
I'm in charge.
Yeah, she can touch it, but not while it's floating so elegantly.
Yeah, you're right.
And
Sometimes
it's,
I get very eager because these moments
Really?
they
Yes.
it just passes so quickly and it's so beautiful. Yeah. Can we stop? Is that still possible? And go back a little? Even sorry, I just saw something very interesting.
Edbridge, can we come to an all stop there, please?
Here it's,
I am.
it's in your camera or?
It is in the lower left angle. I'm currently centering it more. Oh my god, yes. Yes, it's the Staurozoa that Paco and me have been talking about. This,
I see it.
it's special. It's very special.
It's light rosé colored next to the sponge, next to now pretty much in the middle of the picture, the lower part.
We will have a better look at it. We just
Tell
need
me
a-
where it is.
Sorry.
Just
Go ahead.
You confirm that the plan is to start around 1900?
Sorry, say again, please.
You confirm that there is a new plan to start ascending around 1900?
Okay,
Yes.
thank you.
Can you confirm?
Something. Can you confirm?
Yes.
Yeah.
Is that the plan
Wait.
made by the science team?
Yes, I have a nod from the scientists here.
Basically, if you can hear us, Sandra, I mean, we can.
Yeah.
We can.
Yes, we confirm.
Okay, well, I mean, we can do until 2000, we can also do until 1900. I just know that you were wondering to come up earlier. That's totally fine. If you want to do that.
Let's do 1900.
1900 is fine.
Okay.
We'll have a couple of zoom ins before.
Ja, mach was.
Wir bewegen uns ja noch.
Ja, ist ja in Ordnung. Alles gut.
I want to go out.
Ja.
I want to get
Yeah,
this.
you do the driving.
Yeah.
I'm in the back seat now.
You caused all this plume, didn't
Yeah,
you?
yeah. That was all me. That was all me.
I knew it.
Yes.
Have you locked it?
I have indeed not done that. I will even make an important in there.
We will just wait until this has settled.
Yes. This will just take some time. The ones that have watched this for some time know already.
Jenny, what is it that we will be looking at?
It's this Staurozoa, which is in the same group as jellyfish. It is a Staurozoan jellyfish, and it's benthic, so it's attached to a rock.
Which is very unusual in that group,
It's
because
super
normally
unusual.
they swim
Yeah.
through the water column.
Yes. So
And often also mid-water, and now we've seen quite a few of the red ones
Yeah.
closer to the sea floor.
yes. And they're incredibly beautiful. They're like bell-shaped. You will see in a bit.
Hopefully.
And we have seen one or two specimens in a previous dive where we had a similar habitat. And Paco and me were talking about this earlier today, that we were sort of expecting to, or we were hoping to find another one. And we haven't seen any yet, but now we found one finally. Because it is a very suitable habitat for them to settle. They are not that common, so we don't really spot them that often. Now we're slowly starting to see things again. That is just so lovely.
Slowly. So.
Feel the stars already waving?
Yeah, they're all waving. I think it was up there, but I have to look. I lost it a bit now. I really want to see it again. What's happening?
It's me.
Okay, no worries.
The umbilical came off. No.
Oh. Do you know where is it?
Now we're above it, so we have to move back a little bit.
Back to the left.
No.
Yeah.
Now it should be straight ahead of us, but we can't see.
Let's just hang in here for a bit.
Do you want to?
Yes. It is a special animal. We should capture it. Let's just give this 10 more minutes to settle, and if it doesn't get better, I agree that we abort, but I would really love to try.
We can also see that quite some mud is stuck to the RV itself, and that is slowly taken off. Can you see?
Yeah.
Fixed to the mesh.
Yeah.
So we're causing our own.
Yeah, we do. So let's look for it again. It was on one of these rocks, and it was on top. May I drive?
And slightly rosé colored.
Yeah.
Of course.
And
It was more on that side.
yeah, it was. Sorry for the bad cam steering here now.
Can you see if you can look it?
Sorry?
No, just see
I
if
can't
they
see
can
it.
find it.
I can't find it. I have the slight feeling that we have to move a bit more starboard actually to see it. But maybe let me look again before we start moving. It's not on this rock. It was a different one. Sorry.
Sorry.
Let's try and peek a bit towards the starboard. Yeah, I think it might be actually
that
right
one.
behind that arm. It could be.
I'm gonna turn on to the port.
Yeah.
You know what? This is perfect for this shot. For the lights.
Yeah, let us turn first.
Sure, sure. One thing after the other. But this is really pristine. It is just really tough to hover and like really find a single spot and sit and wait. It is very difficult. If we don't manage, it's okay. Then we saw it for a second there. We know it's there. We have logged it.
Have you logged it?
I
so we
logged
know
it.
the exact
I
time?
logged it.
So we can zoom
It
in.
was at
I recommend we
Yeah.
We fly on.
Yeah, it's alright. It's completely fine.
Okay.
We gave it a try.
It might be just
One
behind us.
last try.
Okay.
Okay. No.
And the other one that you were saying?
Which one? Up ahead. It was on one of the small ones. Sorry for the zooming in and out. A bit crazy here now.
We'll have an eye out. It won't be sitting there by itself.
No, no, no. There might be
There'll be
others.
others around.
It's okay. Sorry, sometimes it's a bit. It doesn't really. It's okay. We might spot another one.
Sorry, you missed it.
That's absolutely fine. I notified you very late.
No, no. It was. I was hoovering and then some sediment came. And then
It's all
I lost
it's all
my
good.
photograph.
But this is a
Okay.
very beautiful image.
Let's fly on. Thanks a lot. And lasers on, please.
So I just got a notification. So earlier in the dive we saw lots of red jellyfish, like these pink, purple jellyfish, and there are some jellyfish experts at the University of Bergen, and we can at least identify them to family level, which are the, I have to read it, this is a long family name, Rhopalonematidae. Rhopalonematidae. Okay. Now we know that. I will lock this. Thank you. And there goes the pencil.
And fish.
Mm-hmm. Palonematidae.
And more sponges.
Paco, we just found the Staurozoa. We found the Staurozoa.
Cool! But we lost it again.
And then we lost it.
Oh.
Like, I saw it very late, and then we were hovering, and then, you know, it was a bit,
No.
there is,
Cool. That means they're there.
so just have a look for the Staurozoa, and it was just at the edge of one big boulder. Maybe we'll see another one, that would be amazing.
Were you stopped or no? You were moving actually.
We tried to stop and go back, but we didn't find it again, so it was a bit,
Do you want to sit here? Well,
No.
No, he's going to sleep.
I'm just explaining the cast.
Yeah.
So they should be
Yeah.
there. But if both of you want to go eat, I can be along here. It's no problem. Because you know it's going to close the kitchen in 20 minutes.
Yeah.
So if you want to grab a bite, I think you should go now.
Oi! Okay. This is this is a swimming
now
feathers that I'm.
we will zoom a
Yeah,
little bit, but we will not move.
No, we are not. Oi! This is so pretty.
This is where all your streams are. Look at that.
Oh wow!
Oh wow!
So we
Wow!
collected a bunch of them before. We slurped them. from another
Colophacus.
Yes.
Okay.
So it's
Annelien told us that you will describe species, right, from the list in there.
Yes.
And this maybe.
Can we stop?
oh,
No, sorry, I said
sorry.
Yeah.
Yeah. This is overwhelming.
Yes.
Just take them.
I'm happy either way, Paco, but I'm also happy to skip dinner and
Sure.
just watch the last
No worries.
quarter hour.
Take some.
Um,
Can you hold it so
Yeah.
I can take pictures?
okay. Now we will. Sorry, Jenny?
No, nothing. I'm just, I did a hand movement because I knew the sediment plume would come and I was about to wave it away, but
Yeah, no, it won't listen to you.
nah,
Sorry.
it's just not.
Yes, exactly. We want to focus on the. On this
Getting
one.
there.
Your camera is lower.
Yes,
Yeah.
sorry. Our camera is lower and therefore we see a lot of sediment. But we have a couple of more cameras on other monitors and we can see it clearing up.
But this swimming feather star was really something.
It distracted us.
Yeah, lots. Completely threw us off guard.
Now,
Is the mark.
I'll have them with tea.
I rock with this and Dory. Tulip sponge.
Look at this beautiful.
you know what's sitting on it?
You know what? This is perfect for the one life
Here is
thing.
an that was on this
Sorry, can we turn
rock.
lasers on,
fell
please?
Okay.
Lasers are on.
Off. And now off,
This one
please.
is again is Andoric.
Okay.
Here is a rock that we try to connect, but it's
Sorry, I wish to do something with the light.
It's not that as hard as you can imagine. There's super small anemone that were like living like one beside the other, like making like lines. It was super beautiful. Another complicata. Then the Caulophacus plus the Crinite.
Mm-hmm.
Crinite was trying to get.
I'm not happy with the light.
A rock with a small complicata and other sponges. This is not the
So we're currently doing some light adjustments because the sponge is so illuminated.
It's so over. Can we do something to not have it
not this
so
one.
overlit right?
It's this one.
right this one.
Okay.
Do you want to try that one?
Yeah.
Yeah, it's getting there.
It's got
Thank you.
Crinites also, small sponges. So this is
Thank you.
the one that
Okay.
goes for.
Maybe we could even do the
you-
spotlight
you
thing.
Sample it. And you just give it to.
Leave the port LEDs on.
Okay.
I'm trying to heat also. The port
Oh, there's the
LED.
other jellyfish.
That
Can we, yeah.
crust.
Can we try it? Yes, yes, yes.
This is
much
much
better.
better.
Isn't
Yeah,
The other setting was much
Yeah,
better.
that's me.
the one before.
There's a small glass sponge and a small round thing.
Those are the amphibians.
Thank you.
Are you glad?
Oh, no, I think it's brilliant.
Yeah, absolutely. Just stay.
Sorry, but I was a
No,
bit.
I
There is so much going on, and
Do you want less light or it's okay?
No, please leave them as dark as that.
this is really beautiful.
LEDs at the ground. The ones they're going to fight for is Europe again. I'm not sure the other day when they support LEDs. So the most important one is one.
Okay.
Beautiful one.
Yeah.
You can see what taking it up.
Maybe I would like to need some.
Also clap.
Something on the
This is more smooth. I can't. I can't.
Wow.
Hmm?
Your hat or
Yeah, there. So again with this beautiful sponge we're dealing with, right?
Yeah.
That's nice.
You have the experience already.
But you want it. You want it.
Oh, they want to.
Right. I'll give it away.
And guys,
you very much.
Do you want?
No, thanks. I'm good.
Okay, pretty much fly on.
Yeah?
Thanks a lot.
Oh, yeah.
When they come up in 15 minutes, so much going on.
I mean,
There's always you always stop and zoom in. There's always a lot to see when you zoom. So wherever.
Time, you don't get enough, enough, and then you get it, and you're like,
Is this yours, Jenny?
sorry?
Is this yours?
It can stay.
They can stay here.
No, but they have been like fixing this now again.
Okay, what are you hearing? Would you like to dive into it? Because you can, that's
Because
all
then
Will that influence? I don't want to
there's
go.
a side that
Planning to start with.
so when are you going up?
No, I don't want to take this time.
Try that
for
first.
this. I think we've discovered this. Stay
But
here,
we
do more zooms.
did have a plan to go to 8:15.
each.
Yeah. And then we can still continue to 8 p.m. if there's value. That was the plan. And then we'll be at 8 a.m. in the morning at the bottom at Loki's. So it's completely fine to continue until 8 p.m. this evening. Yeah, let me go and check, okay? So if you guys want to continue till 8 p.m. we can.
Yeah, we were just thinking that.
We don't want to take time from the next station. We have covered this. We will always find nice things to look at. We just don't want to take time from the next.
Okay.
We're patient.
Yeah. Well, the plan was we'll go till 8, and then 8 in the morning we'll be at Loki's
Look, there's another beautiful
at the bottom.
If that
And that's-
works, then we are happy to.
That works.
We are happy to continue.
Then I just misunderstood.
Because Daniel was sounding like you wanted.
Yeah, but then I just misinterpreted.
Yeah, no, let me go and check. Because if you're happy to keep diving until 8, you can completely continue until 8. Okay?
I'm happy with
That would put us at a good time in the morning to be Loki's. It's all good.
Absolutely fine. You just tell us whatever suits best. Overall plan, and we are happy to do
I
whatever.
mean, it's only in an hour.
It's only one.
Yeah,
It's only
it's
1
just
hour
1
more.
hour,
Well, let's have another Zoom then.
No, but we are very happy to
Continue forward.
Yes.
Okay, let me check. Why not?
I will go.
Grab something to eat.
Huh?
And grab something to eat.
Yes, and
then we will take turns and have a look.
yeah, and I will let him know
Yeah,
that
ready
you
now?
have
You ready? Moving off.
We are happy to fly on. Thank
Bridge
you.
RV, can we continue the move, please?
Rob, continue to move 310 at point 2.
Lovely job, cheers.
Jenny, maybe it's safe.
Yeah.
That's okay. Thank you. You're great. You're the smartest. Nein, das wird. Danke. Paco, we still have 1 box left.
Yeah, we do.
We have another
And
hour.
two
And we have
and 3
another
slurps.
hour, and you have a precious object.
And two slurps for your amphipods.
That's fine.
Yeah, I know.
That?
Yeah, because I will have to take care of them.
Okay.
I've been in charge now.
What's the other one? Jellyfish?
Yeah.
Do you want to swap? Shall I log? Whatever you prefer.
No, I'm happy to log. Maybe I didn't put it in the. Yes, there it is.
Minus 3 times square root of evil.
Oh, soon at 2,000 meters.
Just through 2,100.
Mm-hmm.
Look at those. What's this?
I think it's a carnivorous sponge. Cladorhiza.
Look at that.
Mm-hmm.
Another flying one. I love how they swim.
Another carnivorous sponge.
Is it going down on the
Yes.
other side? Okay. Are we on a, are we
We're
on
at
the
the
slope?
slope at the moment, and we get over the crest here.
Mhm.
It'll probably be downhill because if you have a look at the terrain up there, it looks just like that. Not rocky, not nothing at the moment.
Because then we'll stop the dive. If it goes downhill,
Mm.
then it'll
Okay.
be hard to see.
Yeah.
Do you want to be more on the center or to look at one of the
I would
sides?
love to go a bit
Yeah.
more to the right,
the center.
and then
Okay.
actually to stop really in front of another boulder, or so we can focus on some small
Just come up,
parts.
go forward.
Has the time been changed for recovery?
Not really.
1 minute was 19.
Like around 8. Taking off? Or have you?
I saw
We
it 7
stopped.
o'clock.
Okay,
Okay.
stop now.
Okay.
Stop now. We have one more zoom, and then we come up.
Yeah.
We'll have one close call, and then.
That's fine. No, no, I can stay here all night if you want to.
Are you sure?
Yeah.
Some work tomorrow. anywhere here is fine to stop. I would just love to look into the details. Can we stop here?
Yeah.
Thank you.
Bridge, RV, can we come to an all stop there, please? Thank you.
Okay.
Any treasures? Any treasures?
Okay.
We're stopping now.
Yeah.
Because it's going down.
Unfortunately,
So?
it's
It's all
decision
good.
was made, so.
Yeah, no, all good.
Okay.
All good, because it's going downhill anyway, so we won't be able to look good view any. We just at one last zoom. And then
Just one more, please. Just one more. Please, yeah. I'm all right.
biting my tongue not to comment here.
Couple of hours, about an hour and a half, an hour 40 come up.
About that, yeah.
Okay.
And then another hour to decant all your side samples.
Last pictures here from
Decamping
Hans
the
Treasure.
All of this luggage.
Yeah.
2,118 meters. Ready to say goodbye?
Not without completing
Stop.
the transit form.
Absolutely not.
Getting into a bit of luck, isn't it?
Ready to come up? Yes, we are.
What about
Oh,
your
sorry.
HD?
Sorry,
I'm going to use your HD video.
we should stop it.
Yeah.
Yeah.
It's up to you. Is that what you normally do?
Yeah.
Yeah.
We stop
Okay, so
recording.
you gotta stop it.
Bye.
Sorry?
I said this bye to our potential
Oh.
viewers. Do you mind turning it on?
Yeah, upstairs, yeah.
No, I thought you could do it here.
I can. I will try and do it.
Stopping.
Stop
3,
now.
2, 1. Slow.
Yeah.
I think it's pressing on these two.
We'll
But,
go find out. I don't
yeah.
know.
Yeah, take it out.
I will find out.
No. Thank you. Grant? You want this?
Yeah. Bridge RV, that's us all finished here on the bottom for today's show. We are going to start to recover back the deck, let's say an hour and a half there-ish, Basil, okay? We'll see you later.
And hour and a half.
Patty. So, thank you.
An hour and a half.
Ish.
Yeah. Thank you very much.
And then you
Thank
can
you.
Pleasant as always.
then you can help yourself to the samples.
Yeah. And I got one more. Help myself to some dinner maybe before and then.
Okay.
Do you mind if I go to
No.
dinner and you
Yeah.
No
do
problem.
the final saving? as
Yeah,
well?
yeah, yeah.
You've eaten.
I've eaten. No problem.
Okay.